I think it'll be a big number.
And I'm seeing the bottom coming into sight, so here we go for dive 10, the last dive of Voyage to the Ridge 2. Will be really exciting, I hope. And just remember, everybody, that Voyage to the Ridge 3 starts next week, and they'll be headed south, and I totally expect them to be doing some hydrothermal vent dives.
Yeah, definitely. You know, they're going to go to Lost City on the Atlantis Massif, so Voyage to the Ridge 3, return to the ridge. Yeah, and they're going to get down into the Cane Fracture Zone, so they'll see one of these really, really large fracture zone systems that we had hoped to dive on earlier in the Charlie Gibbs, but got delayed out due to issues. And so, yeah, it'll be a beautiful, amazing expedition. Probably not as cool as our expedition.
Well, we have a very cool expedition.
Because we are so cool in ourselves.
I think the gauntlet has been thrown.
Yeah.
Deb, Joanna, good luck.
Bring it.
It's clearly field season. I have an email here from Tim Shank who says he's sorry to be out of touch with our crews because he's on Atlantis diving into Puerto Rico Trench. So we're working all over the Atlantic. Go ahead, pilot. We sure do. I'm all set and ready. I think video's ready. We're just doing a water sample. So. Okay, then, let's have a look around. I saw a couple of small fish, but our initial view of the bottom here, we're at 1,921 meters. I see a fair amount of sediment, and I'm not seeing any hard returns right in front of us, so let's move on up the slope here and see what we see. Not seeing any holes here, Ashton.
Holes. I see actually little, looks like bioturbation mounds. Maybe some type of little critter living in the sediment, some type of little fauna.
Yeah, there's plenty of that. Crustaceans and worms, especially down here, will dig burrows. What do we have here? A couple of holothurians, perhaps? Sea cucumbers? No, that's a sponge. Well, how about that?
We are ready?
That sponge, I assume, is on a little bit of rock outcrop that's attached. It looks to me like a glass sponge. What I don't recognize is the color. Usually these sorts of sponges down here are white or have no color, and what you're seeing actually are the silica spicules inside the tissue. So this is kind of interesting. We'll see if any of the scientists who are communicating via our chat room are sponge experts and can tell us a little bit about this. Because we may have a number of new folks joining us today, I'll go through some stuff that maybe is repetitive from earlier dives. But so a sponge is a suspension feeding animal, a filter feeding animal. Basically has a series of canals in the walls there that are lined with cells, each of which have flagella that beat, little whip-like structures that creates water movement that gets pushed out into that large opening you see. Moving the water out creates suction that pulls water in through the side, and then they feed on very small particles like bacteria. That's good, pilot. I noticed another one. We don't need to zoom at it, but I'm just saying this for the folks on shore. Another one up to the left. And so we have a couple of these morphs of sponges. Yeah, the other one looks tipped over. So that's kind of interesting. First time I've seen that particular one. I don't think, have we been at 1,900 meters yet so far in this expedition? I don't know that we have. Certainly not on the Azores Plateau. This will be not on us.
Not on the plateau for sure.
Mike Vecchione is noting the Lebensspuren on the sediment surface, and this is a term that means life traces, and it's referring to the trails that you see in the sediments, even some of the mounds, that are an indication that some organism has been here and has modified the surface. And so even if we don't see the organism, we see evidence for it. Now, that might be a snailfish. I wonder. Liparidae. Very bulbous head. If it is, I don't think we've seen one on this expedition, so it's great to have this view. But I did bring them up at one point. I noted that just recently in my lab we've sequenced some eggs that we found in a cluster inside a deep sea sponge, and it turned out to have been laid by a fish, by a snailfish. So could be something like that. Thank you. Good view. Good catch, pilot. Manuela says they're on Alpha Seamount. ALFA, so we'll have to check out where that is. And so that is assuredly a sea cucumber on the right side. And this is one of the animals that does create this Lebensspuren, these trails. So sea cucumber is an echinoderm. It's related to your common sea star, but you'll see it has a very different morphology. It's a deposit feeder, which means it moves along the bottom and ingests the sediment on the bottom. Or it might even be an acorn worm next to it on its right. We'll see that in a second. No, that's something else. Yeah, so this is definitely a sea cucumber. Those tentacular-like structures that you see on the upper surface, those are modified tube feet. And this is a kind of a flattened one. Actually, those piles that you see in the front there are fecal tubes, and the white that you see on the inside is the intestine just packed with sediments. And so it really just munches the sediment, ingests it, and then any organics that are in the sediment get digested. So it has a very long and twisted intestine that allows for a long residence time of those sediments on the inside of the gut. And that allows for maximum digestion of any energy that's in that water. Excuse me, that's in that sediment. You can see it's just a little above neutrally buoyant, meaning it's only lightly on the seafloor. And I imagine with a good push from the ROV thrusters, it would probably drift up into the water column. And very often we see these sea cucumbers kind of flapping their body and moving through the water column in order to move to a new position to feed. So this is kind of funny. I'm trying to determine what is the head end. I believe it's the left side. And I'm looking to see if it has any of the modified oral podia. What that means is tube feet that are modified into little grasping structures. It's kind of like a little fist that can grab some of that sediment and then move that into the mouth. That's good for science.
And just to give some broader geological and geophysical context, so we've been diving on the Mid-Atlantic Ridge, as we talked about during our overview of the work we've done over the last 10 days. And as we've come south, we've come up onto the Azores Plateau. And that plateau is caused by what we think is a hotspot. So it's caused by an anomaly in upwelling of mantle material coming from the deep mantle. Now, the oceanic crust we're on from Mid-Atlantic Ridge volcanism is quite young. It's probably still 1 to 2 million years old. It's unclear to me how old the area of the hotspot of the Azores volcanism that we're on. In general, it's thought that Azores hotspot volcanism or volcanism that caused the Azores Plateau, this bathymetric high, this swell in the elevation of the seafloor throughout the Azores, probably started about 35, 36 million years ago. There was a change about 10 million years ago that might have increased that. And the Azore Islands are mainly caused by this hotspot feature, but it's in this interesting area where we have the confluence of both this hotspot volcanism, but also mid-ocean ridge volcanism. And where we are now, we're still on, I believe, the North American plate side of this junction between the North American plate, the Eurasian plate, which is separated by this northern segment of the Mid-Atlantic Ridge that we've been diving on, and then also the African plate. Now those three form what we call a ridge-ridge-ridge triple junction, with two of those ridges, two of those R's, being segments of the Mid-Atlantic Ridge, and then another one of those segments being the Tercera Ridge. Now our next expedition leg, the one that Deb Glickson will be the geology co-lead on, and hopefully she'll talk more about this, that'll be on a segment of the Mid-Atlantic Ridge that separates the North American plate from the African plate. So really, really interesting geological context in terms of what's going on over the entire area. The two islands that we were near the other day, Flores and Corvo, those are the only two islands of the Azores archipelago that are on the North American plate. The other Azore islands are kind of segmented between this area of confluence of the African plate and the Eurasian plate through the Tercera Ridge. Now, one of the reasons this is important from a geological context is it's one of the inputs in terms of trying to estimate what we might see on the seafloor before we dive there. The age of the crust is really important in kind of giving us a first-hand guess on the amount of sediment we might see. So, you know, a general rule is we might have 10 meters of kind of calcareous sediment accumulation per million years of age of the crust. And that's kind of using just an average model of currents and precipitation, and then marine snow, detritus falling from primary productors. But as we've seen, that estimate is sometimes wildly inaccurate or not really well hold, like on the axial volcanic ridge, which was a really young feature but heavily sedimented. So really dependent on how much productivity is going on.
Ground truthing is everything.
Yeah.
It's really valuable to get your eyes down here. Did I hear somebody on shore?
I thought I heard a click in.
I did hear back from our colleagues on the Pelagia, I believe was the ship. Yep, the Pelagia. They're at about 37 degrees, 45 minutes. So they're at least about a degree to the south of us. Well, that's what, about 60 nautical miles to the south. I don't know what the longitude is. But as might be expected when we are, let's see, we're at 1,917 meters. We have a signal that's being brought up on this cable to the ship. We are then beaming it to the satellite. It's going to shore, coming back to a satellite, then going back down to a ship onto their monitor. They say it's a little bit hard to see the sponges, so they can't tell me about the sponges.
No.
Understood, Manuel. It's even amazing you're seeing anything.
And so this looks like kind of basaltic talus and rubble, which isn't unexpected. You know, we're not at the foot, but we're, you know, several hundred meters up this side of this faulted feature. This it's unclear to me exactly what type of fault it is, but it looks like some type of normal faulting process. So this is similar to what we've seen before, where we get all this basaltic talus.
It's funny because I didn't notice it. I only saw the sponge.
I know. It's okay.
So this is an encrusting sponge. It's kind of interesting. I don't recognize this one either. This is the second sponge I've seen that it's like kind of foreign to me. You see lots of openings that are allowing for water to flow into that sponge and be ejected out of that sponge at a higher rate of flow. There's a very interesting set of physical principles that guide this sponge. Sorry, I'm hesitating as we're getting in closer here because the smaller holes, yeah, those smaller holes almost make me think that this is some kind of hydrocoral. I'm sure it's not. I'm sure it's a sponge, but it's just an interesting morphology. Anyway, there's an interesting physics to the size of basically the various pipes and openings in a sponge that dictate the rate of flow of water into, through, and out of a sponge. And the sort of genius part is that you have a medium speed coming into the sponge, and then because you have so many of what are called choanocyte chambers, the chambers where the cells actually pick out the food, the total cross-sectional area of that is really large. The end result of that is that the water's moving most slowly through the part where they can grab the food, and then the cross-sectional area of the outlets is really small, like a nozzle, and so the water's actually moving really fast there. Very cool.
So, classic Bernoulli's principle.
Yeah.
With the force of ejection being dependent on the cross-sectional area of the port.
Exactly that. Exactly that. And it operates at those three different scales in the sponge. Wide cross-sectional area, medium, and then small. So right here, anyway, there isn't a whole lot of suspension feeders that are growing on this this first exposed rock that we've seen in the morning. A couple of sponges, but I'm not seeing a whole lot of corals and larger sponges, and I'm not sure yet if that's going to be a function of food input, stability of this slope, or we're in a bit of a current shadow. The two red dots that you see in the center of the screen, those are lasers that are mounted on the camera, and they are calibrated to be 10 centimeters across, so we can get some sense of spatial scale that we're looking at and size of organisms and features and rocks and so on.
Yeah, not just organisms.
Not just
well,
organisms.
I want to know how big the rocks are.
I'm sure you do. You want the biggest rocks.
No. Anything bigger than about 2 fists probably is just, yeah, overcompensating.
On the back side of, let's look at the sponge first, but on the back side there might be a snail we can have a quick look at. Great. Yeah, that's a dead, so on the lower right of the screen there are two dead sponges, one that's a stiff plate, and I'm wondering if this thing we're zooming in on now is a live version of that plate-like sponge that you were just pointing at. Caitlin has noticed a cup coral. Oh, this is very cool. I feel like I'm going to be seeing a lot today. This is the first time I've ever seen this sponge. This must be a bryozoan. What a beautiful net-like texture to this colony. Below it is definitely a sponge, that sort of frilly thing you're seeing. But this lace-like lattice belongs to a different phylum, the Bryozoa. It's another group of filter-feeding organisms. And this would be a colony where there would be literally hundreds, if not thousands, of individual feeding structures called zooids or polyps. You can see that this is attached in multiple places along that rock. And one of the things I'm looking for is at the close edge. I want to see if I can see any extended feeding structures. There'll be little horseshoe-shaped tentacles. Oh, and there's an isopod on the far left.
Is that just a curved sheet, or is that three-dimensional?
That's three-dimensional.
But I mean, it does. Is it a sheet, or is it a ovoid?
Yeah, no, it's a flattened plate.
Yep.
And so it's probably maybe 1.5 millimeters to 2 millimeters thick.
Oh, wow.
Yeah, so it's a pretty thin sheet that you're seeing. So it's almost two-dimensional. This is one of our best views of an isopod, certainly on this expedition. We've seen lots of amphipods, which are a close cousin. You can see the very long legs on this isopod. If you've seen any roly-polies. Oh, and there's an amphipod swimming. So that's two of the most common paracarids crustaceans that you'll see in the deep sea, showing how different they are. Anyway, if you've seen roly-polies, this is a deep sea cousin of the roly-poly. It's also an isopod. And this thing is really cool when it's in the water column, when it's swimming. It uses swimming paddles that are tucked up on the right side there, which is the abdomen, and it sort of trails those very long legs behind it. So sometimes it looks like a squid or a jellyfish in the water column because it looks like these very long tentacles extending from it, but it's actually the legs. There you see the amphipod. You can see that most of these animals have some amount of lipid in their bodies, so they have some ability to be neutrally buoyant. So that one looks pretty buoyant, but is using its swimming legs just to sort of paddle along. Very nice. There's a little cupcake-shaped sponge hanging from the underside of that rock. Then all this other sort of brown stuff is probably foraminiferal in nature or small crustaceans that are building little mud sticks that they sit on. That's good for science, thank you. So Manuela Ramos is sharing that those sponges might be, that we saw earlier, the brown ones, Geodia megastorella or Craniella longipillis. Yeah, there's that gastropod I thought I saw from a distance. Very nice shell. You see the elongated upper part of the shell. There's a siphon that's sticking out of that. That's telling me that this is a predatory snail, so a fairly advanced snail. There's a thread-legged shrimp. There's a few more of those sponges that we saw earlier. And I even see a black coral in between the sponges and the snail that we're looking at. It's that wiry-like thing that's just above the right laser. There we go. That's our first coral of the day. Probably in the genus Sticopathes. And as it's coming into focus, what you'll see are these elongate tentacles that are sticking out from the side of the central main axis. These are called black corals because the skeleton that they have underneath that tissue is this black color from the antipathin protein that they use to make that skeleton. And all the polyps on this one are on one side, and it's on the underside, so you can't actually see the center of the polyps where the mouth is. Each polyp has 6 tentacles, so if you were to count all the groups of 6, you would basically know how many mouths there are associated with that colony. It's a good view, thank you. And I don't think we need a closer look at the snail. We can move on. Thank you. Several more of these brown tube-like or vase-like sponges. You may have heard earlier that we asked the pilots to trip a Niskin bottle, and that allows us to take a sample of water, and when that sample of water is returned to the surface, it's filtered onto this ultra-fine mesh that is able to capture the DNA that's in the water. And so these various organisms are sloughing off cells and DNA into the water column. We can collect that and then get some kind of analysis of what is the diversity based on the DNA sequences that we see. So we may even be able to possibly identify those sponges if someone has collected them and sequenced them before. Just to the left of the lasers is a pink holothurian sea cucumber. Now it's below the lasers.
The Niskin bottle, originally designed by
Fridtjof, I believe.
Yeah, Nansen in 1894, and later further developed by Shale Niskin in 1966.
Eel-like fish here. This may be a cutthroat eel. And almost invariably when I say that, Mike Vecchione tells me it's a snub-nosed spiny eel. So we'll see what happens this time. Swimming by a stalked crinoid. This one doesn't look like it has the snub nose to me, so I think this is a synaphobranchid eel. Oh, good. Mike agrees that this is a synaphobranchid. It's a pretty common eel-like fish in the deep sea. The mysid shrimp swimming in the foreground. It's interesting. I have not seen very many mysids during this expedition. They're often very common around the corals. We've seen them occasionally, but not a lot. The system that you're watching, the images that you're watching, the video that's being streamed is coming from what is called a dual body system. So the main remotely operated vehicle is the Deep Discoverer, and that's the camera feed that you see on camera 1. And then it is tethered to the Sirius, which you can see a view from the camera on Sirius from camera 2. The Deep Discoverer has its own thrusters and can kind of move around on a, call it a 30-meter leash if you want. But the Sirius is attached to the ship that we're on by a winch cable. And so moving around means moving all 3 vehicles. And so there's a delicate balance, a lot of math that's involved in determining how the ship at the surface moves with respect to surface currents and wind, how the Sirius heading is set and the speed that it moves, and then D2's movements on the seafloor. If we could zoom into the rock, oh, it's a shrimp, I see, okay. Thread leg shrimp on the rock, several more of these sponges. Mike Vecchione notes that the synaphobranchid eels are the first fish to show up at a baited camera. If you put bait down here with a camera associated with it, it's a really good way to find out what are the organisms in the area that respond to food input the most closely, or the most rapidly, is what I meant to say. Yeah, this is a thread-legged shrimp. Very common down here, moving about the seafloor. It's a decapod.
And what does that mean?
That means that on the thorax, that's the part of the body behind the head and before the muscular tail part, the abdomen, that there are 5 pairs of walking legs.
Okay.
Hence the deca, meaning 10. This is a different kind of shrimp. You notice that the legs are not nearly as long. It has, I think, a pretty long spiny rostrum to it. That's the sort of spiky bit that points out between the eyes. And there's another one of the bryozoans. At least I think that's what it is. I still have not seen any of the feeding structures, but that lace-like structure sure looks like bryozoa to me. You notice on the shrimp on top how very, very long those antennae are. They go out in front of the head, and then they curl around and drift up back behind the body. And this would allow it to sense vibrations and movement, potential prey or predators from some distance from its body. This looks to me like a different kind of bryozoan, and the way I'm able to say that from a distance is you notice it looks kind of bumpy along the skeleton. And that's because bryozoans, I said before, they're colonial. Each zooid basically is creating a little case or housing for itself, and they're all interconnected. And on this one, I can definitely see those feeding structures are extended. It's called a lophophore, and that's basically a tentacle, a series of tentacles, a ring of tentacles, and each individual tentacle is ciliated. It's got these little hair-like structures, and they beat. So they're generating their own current flow around them. So in addition to taking advantage of the general water flow, they control the micro movements near the lophophores so they can direct food into the center of that ring of tentacles. Very cool. And a hermit crab further to the left. I can't tell right now if it's got an anemone or a snail shell on its back. A lot of these deep sea crabs have anemones living on their hind parts. So far, as Ashton said, we're seeing more sediment than we are bare rock today. We did pass a stock crinoid back there. Masako Matsumoto, who's a deep sea biologist from Japan, says that may possibly be in the genus Democrinus. We're really fortunate here. We have a number of different scientists who join us online and help us make these observations, tell us what's important to look at, and note, especially if something is so unusual, rare, novel, or important to science in some way that a collection might be appropriate.
So we have the decapods, which were those shrimp.
Yep.
Okay. Do we have what would be an octopod?
That would be an octopus.
Yep.
Interestingly enough, within the cephalopods, the group that the octopus belongs to, there are also decapodiformes, and those are squid. The squid have 10 arms or 8 tentacles and 2 arms, a total of 10. So anything that you can do these multiples of 10.
Do we have decapod anthropods?
Decapod amphipods?
Yeah.
No, amphipods have a different number of legs, so they're crustaceans, but they're not 10-legged.
Do we have dodecapods?
That name rings.
12.
Yeah. Right, but the name is ringing a bell. I don't know if it's for a specific genus of something or if it's a larger group.
I'm going to go all the way up to 20. I just got to keep Googling what the next one is.
But why don't we just jump to centipede and millipede?
Oh, there you go.
There we go. Hundreds of legs and thousands of legs. As I understand it, no millipede that's ever been looked at actually has 1,000 legs.
Oh.
But I think it maybe gets up to 800 or so.
False advertising.
Yeah. There's a good student project.
We
have a shark in the distance.
Yep.
We've been seeing, let's see, we saw a cat shark on one dive, and then we saw the, what were they? Yesterday? There's 2 of them?
Was there a sleeper shark?
Was it a sleeper?
I remember somebody calling that. I don't remember if that was verified.
Oh, looks like claspers underneath. Oh, that's a good view. So my scientist colleagues on shore who know better are making sure that you know that even though it's called the decapod, it has many more than just 10 legs. And so I was very careful to specify that it had 5 pairs of walking legs on the thorax. That's where the name comes from. Yep. But there are actually 8 pairs of legs on the thorax. The first 3 are actually used with the head.
Is that a gulping eel?
I think this is the synaphobranchid eel again, but it was certainly taking a big gulp. So this is another cutthroat eel, Mike Vecchione thinks. And then he identified that shark as Etmoteris, and so that was the one that we saw yesterday. So I believe you're right, that was the sleeper sharks, which is a fairly well-known group of deep-sea sharks here in the North Atlantic. Yeah, Mike's saying the gulper eels there is still opening its mouth. Mike says the gulper eels are quite different, and I do believe their mouth is further set back, and you can see this articulation of the jaw. The fish biologists use the pattern of scales that are on the head, the position of the lateral line, position of the various fins, and so on. This is one of the reasons we like to get a good view of the head. Another one of the sea cucumbers that we imaged earlier, munching its way across the seafloor. Lots and lots of these sponges. I'm seeing 1, 2, 3, 4, 5, 6, at least 7 right in view now. And it does look like there's a certain amount of mounding I don't know, Ashton, if you used the term bioturbation before, but this refers to organisms that dig up the bottom, turn over the sediments, create burrows and mounds and trails and feeding traces and so on.
Yeah, they're also really important. That process of bioturbation and the organisms are really important for the process of diagenesis, the forming of this carbonate hard rock, sedimentary rock, from kind of all the detritus, because you can imagine they go through and they kind of refine it, and that allows for smaller grains that are more mineral rich, that allow for more easily mineral reabsorption and reprecipitation.
This is our Etmoteris shark again. Looks like it's had a few scrapes. You can see a few scars on the backside of the body there. Yeah, I agree. I was looking, I said, well, I think it's all the way forward, but that's the lateral line. Is there like a remora underneath its mouth? Can't quite tell.
Michael is saying a cat shark.
Oh, this is a cat shark. So this is different from the one we just saw. All right, our second shark of the day. Yeah, with the... Caitlin's telling me this is a female cat shark. The last one, yeah, I can see the claspers quite well. Here we don't see claspers. The males would have the claspers. Oh, lantern shark. That was the name, not sleeper shark. Lantern shark. Etmoteris is a lantern shark. That was the last one. Mm-hmm. The first shark that we saw. Jean-Marc Gagnon, who's joining us from the Canadian Museum of Nature in Ottawa, Canada. He's a curator of invertebrates there. Is noting about these mounds that there are many macro and micro invertebrate species that we don't see with this camera. This camera is really good at what we call the megafauna. Megafauna, I think, is usually applied to things larger than 5 centimeters, but I know for sure our cameras can see much smaller than that. But still, his point is well taken. All these things that we can't see with the camera, but it's an important and often undescribed diversity. All the stuff that's living in and on the bottom that's really microscale. And the deeper you go into the deep sea, the more abundant and larger fraction of the community those small things make up. And that has to do with the amount of food that's available to them.
Yeah, we talked about this a few times. Kind of, I categorize it almost as kind of an observer bias, right? We tend to reinforce the things that we look at, and the things that we don't look at or we are unable to look at, kind of skew our perspective. So, we don't have a really necessarily great understanding of kind of that smaller scale, microscale fauna or the infauna just because we don't have the ability to study it.
And it's studied pretty well with what we call box cores. Mm-hmm. These are devices that can be inserted from the deck of a ship into the bottom with some degree of precision. Mm-hmm. Sometimes there are cameras mounted on the box core, and they can be pretty large. The largest ones I've seen are about a quarter square meter.
The largest one I've seen, which was the largest one built in 50 years, was a square meter and a half.
Oh,
yeah.
Jeez, that thing must be heavy.
It was absolutely huge.
Yeah. So that brings up a lot of sediment, I imagine, and so would need a very big crane or A-frame on a ship to handle that. But the nice thing about it is when you have them that size, it leaves a good fraction of the center of the sediment undisturbed. And so then you can do various subcores and learn something about patterns of distribution on a fine scale. But this is the way we've learned a lot about what lives in the sediments. But you don't have the context that we have right now that we're looking at these various fish and invertebrates. What are they doing in their habitat?
And then especially, in the deep ocean, you can really try your best to target the soft sediment, but you never really know, right? Exactly. I've had this experience with gravity coring. So, we have a 5 meter basically steel tube with a several ton steel mast or mass on the head of it, and you drop it off the end of the ship, and then just gravitationally it falls through the water column, and the weight of that head pushes it into the column. But as hard as you might target that soft sediment, sometimes you do hit hard rock, and you can bounce it off or not collect anything.
Sure. So it's so nice to have eyes. So this is a new fish for the morning, and I'm not quite sure. I don't think this is a grenadier. I thought maybe it was maybe a Bethitid, Ophidiid. Oh, okay. Anna Kalakos thinks it is a rat tail. Mike thinks maybe it's a cuskeel. We're not sure because it's facing away from us. We haven't got a good look at the face. Get the position of, where's the mouth, for example. And there it goes. Yep.
So besides our live video feed that we have, where we have a somewhat limited range of visual observation in terms of navigating, and we also have our multi-beam products, our ocean map, we also have a forward-looking sonar on Deep Discoverer, which can send out a pulse of acoustic energy, and anything that's hard or prone that's standing up above the seafloor reflects back, and that allows us to have some 3D spatial awareness of where we might want to head to next. And saying that, it looks pretty bare in the blue view right now.
I'm looking in a couple of different places. I'm watching on shore the various biologists who are trying to make their observations and give me some IDs. I'm also looking at this fish guide that I have on my computer. I'll share the authors with you in just a second to see if we can determine if this is a cuskeel or if this was a grenadier. I have two different opinions in the chat room right now. We've heard several times from Ken Sulak this week, retired from the USGS, expert on deep sea fish, and he has noted several times that when you're on these sedimented bottoms, you tend to see more fish, and that is because you get an increased diversity of the fish. The ones that can feed in the soft sediments have basically a specialized mouth. They're often on the ventral side of the head, so they can feed down below them. In addition to those fish that may be predatory and have their mouth more forward, they're picking zooplankton out of the water column. When we're on a hard substrate, it's a more limited group of fish, more the predator type. I mean, they're all predators, I suppose, if they're picking stuff out of the sediments. But I mean, picking at the zooplankton or picking stuff off corals. Jean-Marc Gagnon is suggesting that the pointed mounds that we're seeing may be the product of bamboo worms. This is a type of annelid worm, polychaete worm. And those worms will be standing upside down and feeding on deep sediments. And so these are their excavation piles, basically, as they feed in the subsurface. Thanks for that, Jean-Marc. Still some debate going on about what that fish was. people didn't recognize the combination of the head plus the fin and the caudal fin. There's a fair amount of rubble that's showing up in the sediments now. I think that's rubble. Can we just get another look at the bottom here? I'm wondering if this is rock outcrop or coral rubble.
I think this is coral rubble.
Yeah.
Yeah.
Has that sort of texture from a
distance.
distance.
And it's way too small to be.
Yeah. But maybe, oh yeah, so that's definitely in the lower right is definitely a cup coral. And there above, so it's coral rubble, but very old coral rubble. It clearly has some encrustation on it.
Yep.
So given, just very ballpark, I'm not trying to put you on the spot here, but given rates of precipitation of the water column, what are we talking about? That's been in place for 100 years or thousands of years?
Oh, I could not speak intelligently to that at all. If we brought up a piece of that coral rubble and we looked at a cross-section of it and we could verify that it is a ferromanganese crust, then we could start estimating based off of the thickness of it, but it's really difficult to do just, visually.
Great. Understood.
It might precipitate at a couple millimeters of iron manganese per million years, so, yeah.
So we were just looking into a sponge there that may be a Geodia sponge, and you can see all these siliceous spines sticking out. The skeleton of a sponge uses a lot of silica to give it the structure. And you notice how brown it was. Oh, there's a different sea cucumber there on the floor, just going out of frame on the left side of center, kind of orangey, and had a very transparent body. Anyway, just to finish the thought on that sponge, all the brown on the outside is all this marine snow, the stuff that's settling down out of the water column and kind of fouling and discoloring the outside of it. But when you peered inside, you saw it was this sort of pure white color. So this is going to be a second species of sea cucumber. This fish, I think, is different. Can you put the lasers on, please? Thank you. This might be a halosaur, different kind of eel. Notice that those pectoral fins are kind of pointing upwards. It seems like it has a broader snout to me. Yeah, I'm getting some agreement on shore that's a halosaur. So Halosauridae is a family of eels. Not sure what species this is. I believe the halosaur translates as salt lizard. Yeah, if you get a good view of the head, that'll be really useful. Okay, we're at a max zoom right there. Yeah, this one doesn't want to show its head. You can see that a lot of these mobile animals move very languidly, very slowly, and they're conserving energy, but they all have the ability to move very quickly. We saw that on one of the earlier fish. As we got close to it, then it had this burst of the tail, and obviously that would be a strategy for avoiding predators. But while they're sort of moving around in search of food, they're trying to conserve energy as much as possible.
Zoom on the head?
As best we could, yep. That's good for science. Thank you. That tail that kind of curves upwards like that is very characteristic of this group of fish. And I think those pectoral fins pointing upwards as well. So before we saw the fish, we were talking about the coral rubble, and I pointed out because it suggests that corals are either living here upslope or were living here. And there's no question that we know that physical conditions and the amount of food can change over a long time scale. So this could have been, let's say, 10,000 years ago, a great environment for these particular corals, but it's not now. Or maybe it is, and they're just upslope. We're on a relatively flat part of the slope. We're going up a much steeper wall, and these may be corals that have died and tumbled to the base of the slope. I get the impression that there's not quite as much marine snow in the water column as there has been on other dives. I know it's it's a hard thing to quantify, but that's my impression.
I'd agree with that assessment. Yeah.
So the marine snow we're talking about are these particulates that are moving through the water column, and they're in part a reflection of the food that is descending through the water column. Depending on currents, it may be stuff that's been resuspended from the bottom, but I suspect this is more material moving through the water column, and ultimately that's the food for a lot of these, or most of these species that live on the sea floor. So if there's not as much in the water column, it means there's not as much food being delivered, and therefore you would expect you wouldn't see as many animals or as large animals. But these sponges that we saw when we first arrived are really abundant, and I think the next time I see one, I'm going to suggest a collection because we're seeing so many of them. So that might be representative of the area. There's a fish almost in the center left of where the lasers are, just hovering above the bottom, I think, unless I'm looking at two shadows. And I'm looking at a rock. But there is a sea cucumber there, so that's something. Yeah, it was just a double rock. I thought it was something floating above the other at the top of the screen. That's what I saw. There you can see the sea cucumber sort of being lifted up a little bit, probably by the current from the ROV. It's a different one than we saw earlier. There you can actually see the tube feet running along the lower part of the flattened, what they call I think the sole, or there's the head end going back down. All right, that's good. Thank you. You can see that the slope is starting to get a little bit steeper. So there is one of those sponges just would be below the lasers on the left. It's a little brown. Yeah, what I'd like is to collect it, actually. If we're in a move, there's so many of them, we can get the next one when we're stable. I would like a collection of the sponge, and I'm saying if we're in a move, we can wait. I've been seeing so many. I'm sure there's going to be more. So, yeah, we'll let the moves play out, and then we'll get the next one. Yeah, we'll have a look at it while we're here, but thank you. So one of the things we also like to do is when we're not certain of the species that we're seeing, and it's something that's really quite common and therefore characteristic of an area, we like to get a sample so that we can say, you know, of the characteristic fauna, here now we can name it. And I've been seeing, you know, literally dozens of these already on this dive. So that's great. So just above this sponge, there's something white growing on a stalk, and it's going to be to the left. And I think it's another tunicate. Just coming into frame now, top center. Well, maybe it's another sponge. Boy, some of this fauna is fooling me. I'm not exactly sure what to make of the white spots that are on. It's growing on an old dead piece of skeleton, and now I'm not sure if it's a tunicate or a sponge. It's just, I feel pretty shameful as an invertebrate zoology professor. I think it's a sponge. Pineapple sponge, says Alan Collins. I don't know if he's saying that with authority or just what it reminds him of. And great. So Alan Collins, who's at the NOAA National Systematics Lab at the Smithsonian, also knows quite a lot about sponges, agrees that that brown sponge that we were just looking at is really unusual and is worthy of a collection. Thank you, Alan. I'm pleased to see there's a number of very talented invertebrate zoologists who are in the chat room, and I'm pleased to see that they are also not sure if we're looking at tunicates or sponges, including that red thing, that red-brown thing. So it's not just me.
And what is the...
Frilly?
Yeah.
Yeah, so those are cup corals, long-dead cup corals that have been... They're encrusted for sure.
Yep.
I agree with you. I don't know what they're encrusted with, if it's manganese or something else, but they've been dead for a very long period of time. Material like this is actually of a lot of interest to paleoceanographers who can look at the stable isotopes of the skeletons. Basically, they can understand how old is the structure from those data, and even more mind-boggling, by looking at the oxygen isotopes, they can learn something about the temperature of the water at the time that the coral skeleton was deposited. And so you can start to learn about, well, what was the water temperature like thousands of years ago, and how has it changed this place?
Well, do we want to sample one of those too if we're here and we're putting down for sample that?
Well, but I can't tell if it's an in-place thing or it's just a tumble. It's the same sort of problem that you have with rocks that have tumbled down. So I would say no.
Yeah.
My experience when I've been to sea with them is what they're looking at is if they see a bunch that are growing on a wall and there's a pile below it, that's fair game.
Yeah.
But this is a sedimented slope, so we don't know.
And you can look around and it looks like there's a lot of rubble in general, so not really sure what's in situ.
So now, Pilot, I realize we've been here for some time. If we're stable, let's go ahead and collect a sponge. If not, let's get one further upslope when you feel comfortable with the positioning. Yeah, it's so small, I think we should suction. Okay, so I think we're going for this one here. And I think we're getting a consensus that this is more likely a tunicate than a sponge, just giving us all that vibe, which would explain why I've never seen this sponge, because it's not a sponge. Yeah, it's on a coral skeleton underneath there, and it's fixed pretty well. I suggest that you stop trying to suck that. Move the suction to the frilly coral just above it at 1 o'clock. We'll take that as one of these fossil coral samples for aging that Ashton noted, and then we'll pick the stick up. Is that possible? Oh, yeah. That little clump. So is that Mini Zeus? Yeah, I'm looking at Mini Zeus just to its right. Thanks. Yeah, right there. No, I'm talking about the black stuff, pilot. The skeleton, the black skeleton, immediately to the right. On Mini Zeus, it's just behind the wrist right now. We usually have, if it's usually broken up. Yeah, good. If it's all attached, don't do it. But usually they're not attached. If it's concerning, if you think it's going to do the hose, let's just stop and, yep. Yeah, it's that cup just above that I'm really looking for. Yep. Don't worry about that piece, that one just to its right, the frilly cup. Yeah, right there. That's the one. It's kind of hooked. I think there's a little branch on it. Can that go in the rock box? Yeah, no, so I was trying to avoid using a jar, and I figured if we already had something, then...
You can put it in the lidded rock box, that would be fine.
Yeah, if it can go in the...
If you can drop it.
The port side rock box, is that possible? Yeah. I see. All right, go ahead, in a bio box. I was just trying to make the best of, since we were sampling something in a canister, I didn't really want to take up a bio box with this. Dumping it back in the seafloor. Thank you. Yeah, no, we got something in there. Yeah, no, I completely understand. That would end our sampling with suction for the day. Yep. Yep. Yeah, I'm still calling it a collection. We got some fossil coral bits and sediment, so I will call that geology, I guess. Thank you. Yeah, no problem. So again, the brown ball is really what I wanted. When I saw that wasn't going into suction, I just thought if that was a pile. So I apologize. There was too much consolidation there, including the brown mass. We're going to get one when we're in a more stable position. So we've now had the ship and Sirius move, so we want to move the Deep 2 Discoverer ROV into a better position. Can we get a look at this? It may not be cooperating. I was going to ask, but the fish had zoomed off. It's kind of settling again. Yeah, and laser's back on, please. Thank you. Yeah, it's somewhere that's not where we are. That's probably the problem, which I completely understand. We're a pretty large vehicle moving through the water with some bright lights. Probably surprising to most of the animals down here. I'm not sure if this is the one that's giving us some problems for ID. I mean, it does kind of look rat-taily like, but it's got a very rounded head. Yeah, so Mike agrees. Rat tail or grenadier. Getting different suggestions here. Rat tail, possibly genus Coryphaenoides. Les Watling is suggesting maybe the round-nosed grenadier. I had thrown out a Bathygatus ID, but I don't see the filaments on the pectoral fin, so I don't think that's actually going to be correct. Oh, if it would just turn just a little bit. No. All right, let's leave it at there. Let's make some way. Yeah, this is I think this is a different eel than we've seen previously. Oh, Mike believes this is another synaphobranchid. So we have been seeing this a few times today, so that's fine. So we're trying to get up this slope to the steeper portion.
ROV nav, could I get range to waypoint 2, please? Two, 6, 8. Wait, what? A distance to waypoint 2 is 2. That's the next waypoint coming up. That the distance to the next waypoint should be less than 80 meters, I thought.
He's looking at the top. You look at waypoint 1, they must have...
Okay, I see. Yeah, No, understood. Yeah, we have an intermediate one on our flater mouse that I was looking at. Thank you.
So we're looking at a holothurian that's swimming in the water column right now. Just released some of its ballast, which would be the sediment that's in its gut. The head end is at the top. And this is a different species. I saw one of these earlier. We didn't get a look at it, but this is at least our third, maybe our fourth different species of holothurian. Some of the tube feet have been fused together on the dorsal margin that's the back into kind of a sail or a fin. You can kind of see that flapping once in a while. That kind of presumably helps push it in the water column. But it'll do this to move away from the sediment, presumably to find a new place to feed, or possibly if there's a predator, it needs to escape it quickly. But most of the life is spent on the seafloor, mouth pressed against the seafloor, feeding on the sediments. It's kind of cool, the transparent body. You can see all the different internal organs. So just a brief recap of what's going on here. We are diving in the deep sea in the mid-Atlantic ridge, and more specifically on the Azores Plateau. We're about 150 kilometers almost due west of Horta, maybe a little, I'm not sure if we're a little south or north, but the island of Fayal in the Azores. We got onto the bottom at 1,921 meters depth, and I have just changed my screen. Let's see, we are currently at 1,860 meters. Sorry, just changing some of the monitors in front of me so I have access to the data. And let's see, we've been on the bottom for about an hour and a half, I think. And we are climbing a ridge, and where we landed was a lower slope, or a less steep slope compared to where we are going. And just ahead of us, this wall should get significantly steeper as we move up and see if we can acquire the crest of the ridge that we're diving on. And what we're seeing on the bottom for the most part is an awful lot of coral rubble. There's a large dead sponge skeleton that we're just passing over there that's draped in sediments of various thicknesses. A little bit earlier we tried to collect a tunicate, somebody's calling like a pineapple tunicate based on its color and its shape. And we couldn't see it because of the sediments, but it was very firmly attached to some coral skeleton branches, or subfossils, I'll call them, of the coral branches. And so it wouldn't release, and we couldn't pick it up. But it's an indication of how deep the sediment is, and it's covering some of what's going on in the bottom. Looks like a different kind of a sponge here. This one's a sponge for sure, maybe in the family Fariidae or Eurydidae, one of those two I would imagine. There's a couple of sea urchins also here on the rock. Maybe we can get a look at the urchin that's to the left of the sponge. That might be interesting. Yeah, some pretty long spines. The brownish one on the right is also a sponge. It's dead, and so the skeleton has all this sediment moving through the water column that's settling on it and is unable to clean it off. Whereas the live sponge, perhaps through the currents that are generated, can keep the surface a little bit cleaner using current flow and the mucus that it produces. There's a little hermit crab on the rock behind that sponge. And that's good for science when video is finished. There you can see our 10 centimeter across lasers, get a sense of the size of that sponge. It's not a very big sponge, maybe 20 centimeters tall. Should be on the order of about 10 inches. Yeah, so we're passing over more of these brownish tunicates. They've been really common since we landed on the seafloor. I thought early on, well, I guess when we looked at it, I said I wasn't sure if it was growing in the sediment or on something hard just below the sediment, and we could see from that one that we collected, it's on a hard substrate, so that tells me that the larvae are looking for something hard to settle on and grow. At least so far, they're definitely the most common fauna that we can see. The sponge coming into view, it's white just to the right of center near the top of the screen. This looks like a polymastia sponge. You'll see it's got a lot of what appear to be spiky bits to them. And I think there are various chimneys or excurrent oscula. We did collect one of these, I think 2 dives ago. Came up in quite good condition. It was remarkably dense. A lot of tissue on the inside, quite heavy. So that is most certainly what this is, although this morphology, the gross morphology, looks different from the one we collected, which had a lot more toothed chimneys, maybe I could say it that way. These things that look like spines, they're not really spines, but the things that look like them had a lot more side ridges on them. I don't know if that means they're different species. I don't know what we call plasticity in biology, the range of variation of morphology of these sponges. But I'm pretty sure it's in the same family of sponges. And I don't know what the biological function of those spikes is, and I don't know if it somehow baffles water flow around it. It's kind of interesting. Thank you for that. See another one of the urchins that we just imaged.
Those spikes are outflow?
Well, that's what I thought, but I don't see any holes in those ones. I'm going to have to go back and look at the one in the lab. There's clearly outflow in that tallest chimney in the center.
And where's inflow?
They're much smaller holes. You usually can't see them.
I guess, from a physics standpoint, it would make sense if there was some type of flow through those spikes, if they, from their structure, right, in terms of the longer they are, the increasing circumferential area they have around the organism, where if they were inflow or outflow and they were really near, right, then you're sampling inflow or outflow around a smaller portion of water, right, so the larger distance you
get
away.
Capturing like a larger volume around you.
Yeah.
And using less tissue to do so. So it's kind of less energy investment, but sampling a greater volume of water. I think, what we're seeing, Ashton, is very different from the previous dives. I don't recall seeing, well, we've seen lots of rubble, lots of coral rubble. I understand that. But we haven't seen so much loose coral rubble that there aren't a lot of other things growing on it. That's what I mean. This is I think so far, the most depauperate dive that we've been on.
Yeah, and I'm curious whether that's that lack of really well fixed hard rock or hard coral, paleocoral exposure. There's not much, at least on those flatter areas that we were at, there wasn't much that was really prone above the seafloor.
So another thing is, so we're diving on the flank of a ridge. This is the long ridge that's oriented more or less north-south, and we're diving on the eastern wall, kind of headed west. And if the dominant current is actually intercepting the west wall facing east, that is, the water flow is coming from the west, then we're in a current shadow. And that would also explain why we aren't seeing much. And in order to answer that particular question, we would need another dive on the west side to see, well, at the same depths, given generally the same slope, do you see a whole different community, which would tell you there's a different controlling factor than just what's on the bottom and the slope of the bottom.
Now we might see some change in diversity and numbers as we go up on the steeper portion of this. So this is much more what we're seeing now, what I expected to see for most of this dive, given that we are on one of these exposed faults that's excising, the upper several hundred meters of oceanic crust of 1 to 2 million year old pillow basalts. And what we're looking at is basically if we took that top few hundred meters of oceanic crust that's formed at the mid-Atlantic ridge through pillow volcanism, eruption of basaltic lavas that form these really beautiful pillow mounds that we saw in some of our earlier dives, and then you basically just removed or uplifted or down dropped the few top couple hundred meters to expose it. That's what we're looking at. So we're looking at a cross section of it. And the pillow structure tends to be hard to see just because of the amount of shear stress that it goes through, and it highly fragments that rock. And then, of course, talus, pieces of basaltic rubble come on top of it. And so maybe as we get more of this hard rock exposure, we'll have more places for some of these sessile fauna to anchor.
We're looking at another sponge and another isopod to its lower left. I think this is similar maybe to the sponge that we imaged earlier that had all the brown. I think we got both of these before, so we don't need to spend a lot of time here, but if you want the image, go for it. So, yep. Isopod crustacean there with very long limbs. Isopods are very common in deep sea, and they're very diverse. There's a superfamily that has probably hundreds of species. What I was going to say about this sponge here, it must be, I suspect it's a younger one because it's not all coated in sediments yet. Earlier we saw one that had quite a lot of sediment on the outside, but was clearly alive on the inside. And not that I see here, thank you. We can continue upslope. Dead center. The little red thing, yeah. I think that might be a thread-legged shrimp. Just off screen to the left, is that what you mean? Yep. There you go. Nematocarcinidae, I believe, is the family. It translates as these thread legs. Again, you can see that really long antennae that can sample out pretty far into the water column. Can't tell if this is a encrusting sponge or an encrusting bryozoan. I think it's probably a bryozoan. I think we noticed something towards the very end of the dive yesterday. Looked a lot like this, and it's kind of interesting because that would, I'm saying bryozoan, I'm sorry, I mean tunicate, an encrusting tunicate. And that's kind of interesting because we think these other brown things are also tunicates, these brown pineapple-shaped structures. And we've collected a couple of tunicates earlier this week on this expedition, which I think is a little bit unusual for the program. Tunicates aren't something we often get to collect. There we go. See that nice blue-purple color? And all this color you're seeing are from pigment molecules, and sometimes they're associated with some interesting bioactivities, so may be of interest to natural products chemists. But this one, I don't know. That actually may be a sponge. Seems like the more dives we do, the less I know. The less I'm able to identify stuff.
I still believe in you.
Thank you. It's just, I don't see the characteristic pattern of zooids that I would expect to see in a tunicate. And I suppose it's possible that all those zooids are somehow housed deeper within the tissue and I can't see the in-current openings. But that's why I think of it, it's a sponge, because in the sponge it's more common that you can't see the openings that bring water in, and you can see a few number of large openings that channel water out. A lot of science is about making observations, asking questions, making predictions, and then making follow-up observations or experiments where necessary to answer those questions. So in that particular case, there's a variety of other things that I could do if we had the time or if we wanted to make a collection in order to ascertain whether that was a sponge or a tunicate. But for now, we'll just take the nice images and hope that the appropriate experts see them and can provide us the identification. So I think this is the lacy bryozoan we've seen a few times today. I'm looking on camera 2, the Sirius camera, and this looks like the biggest outcropping of rock that we've seen today.
Mm-hmm. But
certainly solid.
Hopefully reminiscent of what we've seen on the other ridges in terms of it should look the same.
Yeah, if you go to camera 2, you get a good image of what it looks like for the ROV Deep Discoverer in situ hovering above this wall that we're climbing. Get a sense of scale. The other thing you can see is that the image in that camera is moving up and down, and that's because it's connected to the wire, the winch cable from the ship. And as the ship moves up and down in the swell on the surface of the ocean, that's translated down the wire. But D2 has its own propulsion, and so isn't affected by that heave. And so you get these really nice stable images in the main camera. So if you get seasick stay on camera one.
Have you ever got land sick?
I have gotten, yeah, I mean, not to the point that it's causing me to throw up or anything, but absolutely spinny, dizzy, losing my balance, and especially after a very long cruise, or that cruise has been particularly turbulent, shall we say, at the end with lots of swells. I don't know why it is. Very often when you're coming back from a long cruise, you have a long transit back, you always seem to be beating into the wind and against the swell and the waves. And in those situations, it takes a day or so to clear out. Especially when you're sitting at your desk, you feel like you're swaying back and forth. It should be. Your brain wants to be compensating for that swell that's not there.
Yeah, I remember my first cruise at the University of Hawaii on the Kilo Moana. It was about a month or maybe 5 weeks. First time ever being out at sea on a ship like that for that long. And I walked down the gangplank and I stepped onto dry land and I immediately just started rocking back and forth.
Yeah,
and
it's like grab for something. Yeah. What's happening? Yep. We've actually been quite blessed with the weather for the last few days. Started off with some heavy seas when we were farther north, but the last couple of days have been really quite calm, and we're going to have, I think, a relatively easy transit in just overnight into our port in Porta.
Well, talking about the islands of the Azores, I'm going to give you 3. Uh-oh, do you see something?
Yeah, pilot, if you could look on the upper right corner, there's a small yellow pear-shaped, probably a sponge. Yeah, go ahead.
Are there 2?
There's 2. I'm looking at the one that's below. That upper one is the Geodia-like one.
Well, we've seen a few times, yeah.
Mike Vecchiano says it's easy to ID the things when you don't know that you don't know. Yeah, so this one's on a little stalk. This is a pretty classic glass sponge, I think. Maybe Hyalinnematidae. The glass sponges belong to the group Hexactinellida, and they take on just incredible complexities of architecture of the scaffolding, the skeleton on which the cells are growing. Lots of diversity. If you want to get a good sense of the diversity of glass sponges and some of the things that we see in the deep sea, I encourage you, if you go to the Ocean Explorer website, have a look at the Benthic Animal Guide. I think it might be under the data tab. And then go to the sponges, glass sponges, and then just peruse all the different images of glass sponges there. I think your mind will be blown by some of the crazy shapes and sizes. So here you can see through the tissue, and you're seeing that silica framework on the inside of the sponge. And the way it's connected to the rock is also through a whole lot of silica fibers that are forming that little stalk. There you go. You see the attachment to the rock point. And some of these are able to extend those fibers into sediment and basically root into the sediment. Yeah, that's great. That's a really nice image. And you can contrast that as you just look up above. There's another sponge of a different species, and you see this is the one that I've said seems to accumulate more of the sediments in the water column, so it looks like it's got a brown rind on the outside, but you can see that white tissue on the inside. And you see lots of these silica spicules extending from the margin. I don't know if this is just a young stage of a Pheronema sponge, the bird's nest sponge, or if this is something completely different. I am not a sponge biologist, so I can't ID these quickly on site. Really nice view on the inside. And again, the opening that you're looking at is the excurrent opening. So this is where water would be leaving the sponge after it has been filtered for food particles. All right, good stuff. And I think there's another one up above it to the right. So it's interesting, the sponges and the tunicates are ruling here so far on our dive, and my impression from what I've seen in the various deep sea dives that I've been doing in my career is that that's often a factor when you do have relatively low food input or low currents. They must have a lower metabolic need than some of these other species, and so are able to withstand the lower amounts of food that are available. There's certainly no shortage of rock to settle on, so we can't say that the substrate isn't stable enough to support corals growing over the long term. That's not the problem. Can we get a zoom on the blue-purple thing that just came into frame, upper left? Thank you. Okay, this one, I was about to say this one must be a tunicate. I'm going to reserve judgment on that. No, that's a sponge. You look inside there, that's a sponge. I think that last one that we saw was a sponge, but it's the same thing. I wish these were growing on something that was a little easier to pick up. I'll bet that would be a really interesting one. Thank you. No, we're okay on that. Thanks. I think it's the same one that we were imaging earlier, just it's a little higher, so it's easier to light. One of the thread-legged shrimp, the red thing in the back. Oh, there's our first Galatheid squat lobster. He's watching over this purple sponge. He was just to its right.
And so if you are just joining us, we are currently on our last dive of this expedition. We've been spending the last week and a half diving around the mid-Atlantic ridge north of the Azores, slowly working our way down into the Azores Plateau, or onto the Azores Plateau. Today we're diving about 150 kilometers more or less west of Horta. We're at this really interesting tectonic area where we're just within a few dozen kilometers of a triple junction. And what I mean by a triple junction is a place where three tectonic plates are separated. So when we were diving on the mid-Atlantic ridge north of here, we were diving on what we call a divergent plate boundary. It's a mid-ocean ridge where new oceanic crust is created, and that specifically separates the North American plate from the Eurasian plate. Now where we are now, we're in this interesting area where the mid-Atlantic ridge, one of these mid-ocean ridge, bifurcates, and it kind of separates into two segments. The segment that we've been on, and then another segment south of us. And along with that, there's another rift system called the Tercera Ridge, which intersects that bifurcation in the mid-Atlantic ridge. So we have two segments of the Mid-Atlantic and then the Tercera. And they're all spreading centers. They're all places where new oceanic crust is created. And that place where they join, we call a ridge-ridge-ridge triple junction. And so where we were before, we were on the North American plate and kind of jumping back and forth between the North American and the Eurasian plate. Now at this triple junction, we also have the African plate south of us. So we're in this really unique area. The place we dove on yesterday was near the islands of Flores and Corvo, and those are the only two Azorean islands that are on the North American plate. Where we are now, we're still on the North American plate. As we move in our transit into Port and Horta tomorrow, we'll be getting on to this kind of segmentation between the Eurasian plate and the African plate. Now, not to complicate it, but there's also the thought that there's actually another plate involved. There's another one of these tectonic plates involved called the Azores microplate. And so that might be its own little special, unique tectonic plate that's moving independently of those other three plates. So that basically is bound by the Tercera Ridge north of us and the East Azores fracture zone south of us. And a fracture zone is just one of these places where segmentation in one of these mid-ocean ridge systems is accommodated. So we dove on one of those a couple days ago, the Kurchatov fracture zone, where there was an offset east to west in the north-south trend of the Mid-Atlantic Ridge. Now, the oceanic crust that we're diving on, the stuff that you'd likely see here, is probably 1 to 2 million years old, and we can estimate that kind of tectonically by looking at the spreading rate, or the rate that new oceanic crust is created at the Mid-Atlantic Ridge, which is on the order of 2 to 4 millimeters per year, and then the distance we are from that ridge. So that gives us an estimate of 1 to 2 million years old. Now, the Azores Islands are formed through what we call hotspot volcanism, and that is a different process than the mid-ocean ridge volcanism that we've been seeing on our dives previous. Mid-ocean ridge volcanism here extends all the way from where we are now all the way to the mainland of the United States and all the way to the mainland of Europe. So we're talking, 60, 70, 80 million years of oceanic crust. The Azores hotspot is thought to have developed probably maybe around 36 million years ago. And here's a beautiful little, waiting for my shore-based scientist to tell me what it is so I can finish that sentence. Beautiful Q-tip, cotton ball. I can't tell. It's so encrusted. I think it looks like a little. I think it's a sponge. I can't tell if it's a glass sponge. It's one of those ones that starts with the word demo. Yep. Mysid.
The mysid must have photobombed. That would have been a little possum shrimp.
Yeah. I mean, that's just so. It's interesting in how encrusted it is.
Yeah, it's been there a long time, I think. It's interesting to me that how many of these sponges look like tunicates when you approach them. Oh, nice fish. Did you already do the fish?
No, the fish just came into view.
Yeah. That's a good one.
To get a nice beauty mugshot.
Yep. Again, very slow moving, languid, chill.
Mm-hmm.
Let's see.
I'm going to guess it's a cusk eel.
Okay. I think that's an excellent guess.
And I know I'm right in that guess because it wasn't really a guess. I saw Michael Vecchione put it in the chat.
Yep.
Our wonderful shore-based scientists who we communicate through chat and who also call in sometimes.
Yep. It's passing over an urchin, another sponge, a little stock sponge back there.
These fish that are this deep, what are they usually consuming, and why do they move so slow?
Well, it's a good question about what these fish are consuming. So there's lots of different fish that have different diets, and I'm not an ichthyologist, so I'm not certain. This one I see the mouth is kind of forward-facing, not downward-facing, so I suspect this is not sucking things out of the bottom. I think that's probably what the rat tails are doing. I would imagine that this is probably feeding on things like the shrimp and amphipods and mysids that we're seeing, all the crustaceans. And there's a question about why are they moving so slowly here? And one of the reasons is, it's cold down here, so it's about 4.6 degrees Celsius is the water temperature. And there's not a lot of food, so you want to conserve your energy. With a cold temperature like that, it probably has a relatively low metabolism. It's basal metabolic rate, that's something we hear about. So it doesn't, if you have if there's not much food available, one way to deal with that is you have a very low energy output, conserve what energy you have. And so if you just kind of don't use your muscles too much and just kind of drift along, then you're saving that energy for when it's needed, when you sense your prey and you need to go after it, or when you sense your predator and you need to get away from it. One thing I'll notice is there's a couple of barbels hanging down from the fin there, the little filaments, and those are probably sensory. So they're sort of feeling around things on the bottom, and that might be one way that it's detecting its prey. Behind it is passing over a rock, and there's some stick-like structures extending up. I think that's probably going to be a carnivorous sponge. I'm getting a question here, are cusk eels edible? I'm not sure. There's a suggestion here from Tina. It's above the urchin, directly above the urchin against the rock. Let's start with the snap soon. I'm not sure if it's a carnivorous sponge or if it's just some filaments that are hanging off the rock. They're filaments, but I don't know filaments of what exactly. Huh. Oh, it's way out there. Yeah. Yeah, if you're going that way, if it's not on your way, don't worry about it. Well, at first I thought these might be filaments of carnivorous sponges, and that's not the case. I'm not sure what they're filaments of. Yeah, that's fine. It's not something we want to collect or anything like that. Thanks.
Now, I'm going to guess for most of the dive, what we're probably going to be seeing is we're going to see more of these highly fragmented pillow basalts. This entire kind of exposed fault structure that we're on, which we started about maybe 2/3 of the way up, the whole thing's about 600 or 700 meters tall, and that probably entire thing is made up of these pillow basalts, and that's not unsurprising. The top portion of the oceanic crust, the top 1 to 2 kilometers, are made up of pillow basalts from small pillow volcanoes that are built on top of each other at the mid-Atlantic ridge, where we were diving on earlier. I think that was our fourth dive or third dive on the axial volcanic ridge through the same type of processes. Now, underneath those pillow basalts are what we call dikes, and these are the same type of basaltic material, but instead of being erupted in the water, they're just emplaced within the crust. They never actually make it to the surface, and that allows them to crystallize and cool slowly and form these really beautiful structures that tend to be very linearly and very linear and vertical. If you've ever been to Devil's Post Hole in the Mammoth area of California, you see the beautiful columnar basalting. Same on Giant's Causeway. Now, beneath those pillow basalts and beneath those dikes, we have kind of the magma reservoirs that feed all these volcanoes. And as they cool slowly, they get into something called gabbro. That's a mineral constituent. It's a mineral assemblage. And then beneath that, we have the upper mantle, which is made up of another mineral called peridotite. One of the ways we know this structure and we know these mineral assemblages is by actually looking at pieces of oceanic crust that have been exhumed through tectonic processes and are now visible on land. And these exposed pieces of oceanic crust that are on land, we've termed ophiolite. And there's two classic ophiolite deposits that people use as analogies for what the oceanic crust looks like. And that's in Cyprus and Oman. I had to get in a quick geophysics geology background before you saw another sponge. So, right?
There's lots of sponges to see here.
I take my break.
That. Yeah, that's good. No, well done. That's good, pilot. Thank you. There's some interest from some of our colleagues on shore in one of these sponges, and I'm just trying to determine which sponge it is. There's a timing issue, a little bit of a lag as we're passing over, and I think it might be the one that's just to the coming into frame on the left, the one that's got the sort of brownish outer appearance and the white center. That's what I'm trying to determine. Need to find an image of this. Yeah, I think that is indeed, that looks a lot like what we're looking at here.
Now I'm going to ask a question, and this isn't just for Scott. This is for anyone. This could be for our video engineers, our video team, our ROV team. What do you think is larger, or place them in the order of biggest to smallest? The state of Rhode Island, the state of Delaware, or all of the Azores Islands exposed? What's biggest, what's smallest?
He said he wasn't asking me, so that's good. I'll pass it on.
Well, that's good. So where do you think the Azores fit into that? You guys are correct. The biggest is Delaware at 2,500 square kilometers, then the Azores Islands, 2,330, and then Rhode Island, Little Rhodey at 1,500 square kilometers.
These guys are good because they also go through the navigation training.
Yeah.
Have a natural instinct.
So our ROV team got that question spot on. But it gives you a kind of a scale perspective. There are 9. Yeah, Hawaii has much more surface area. Can you name the Hawaiian Islands? Just Hawaii. Yeah.
Niihau.
Yep. Yep, that's it. Yep. Good job. Yes. Yeah, Kahulave is not inhabited. Yep. Oh, very good. Oh, the state fish right there. They're all named undersea features, and the ones that do break the surface are named as well.
Loihi.
Well,
On its way.
Well,
On its way.
Well, Luihi is not the name anymore.
Oh, it's not?
No, they changed the name.
Shows how old I am.
This was just recent, within the last year.
Shows how young I am.
Yeah, the new name of Luihi, and excuse my pronunciation, is Kama Ehua a Kana Aloa.
Well, we're certainly scaling a steep slope. Now I'm going to be the one who wants to get to the top.
There you go.
To see if there is a significant difference once we get onto or out of the potential shadow of this wall.
Yeah, naming of geographic features is always a kind of an interesting. It's an interesting story, right? Because we came through as scientists, and we air quotes discovered Loihi. We mapped it, but discovered is unfair because there are actually ancient oral traditions and stories in Hawaii that tell of the submarine volcano that formerly was known as Loihi. And so they knew of its presence, and that there was, a historical and cultural reason to have a different name for it. And so it went through a process of kind of looking at the name and the reasoning behind it.
Lower left. Oh, sorry, I'm looking at a sea star lower left. I don't know what else was seen.
Center was a cookie star.
Oh, I see. Yeah. My apologies. Yeah, thanks, video. I didn't see that one ahead. No, go to the left. Let's go to the left first. Yep, I understand. This one, actually, that's a slime star. It's not a cookie cutter star. Oh, well, maybe it's not either. I don't know. Well, it's certainly a sea star. It's an asteroid. We can all agree on that. I don't think it's like the Terranaster or Mixaster stuff. I just don't see an osculum really on the top unless it's really quite small. The cookie cutter stars, I think, are goniastrids, and they have much thicker plates all around the margin, and this doesn't have that. It reminds me of slime star without a well-developed, whatever they call the dorsal skin. And I prefer, let's just get a quick look at the other one and then move on. We really want to get upslope here, so. Thank you. I just don't want to delay our move upslope is my main priority.
After this, do you want to go eat?
This looks like one we were looking at. I don't remember if it was yesterday or the day before. Chris Ma got on and told us some about this. You see, so sea stars are echinoderms, and that name translates as spiny skin, and it's referring to calcium carbonate, small hard parts that are embedded in the tissue. And in the different echinoderm groups, the sea stars, the sea urchins, the sea cucumbers, the brittle stars, and the crinoids, those ossicles are fused in different ways. And one of the things as you zoom in here, you can kind of see that there's an architecture to the body, and that's these various ossicles, the way they're put together to form those rays or those arms. And then on top of that, you also have a varying degrees of spination. And this particular sea star has an awful lot of spines that are projecting on this dorsal margin of the arms. The mouth for the sea star is always pointed down towards the substrate, and they move around on tube feet. There's basically a hydraulic system of a hydraulic mechanism that can control the tube feet, that they sort of glide over the surface. And there are lots of different species of sea star and lots of different targets for their feeding. I don't remember exactly what that one is feeding on. We do see that there are many coral predators and sponge predators. That one may be non-specific kind of detritivore, omnivore, you know, whatever it encounters.
And so our fearless biology lead, Scott France, is going to lunch. We are undergoing an ROV team swap. The loud siren in the background was the test of the ship's morning alarm. Our illustrious video editor, Caitlin, has been replaced by our other illustrious video editor, Anna. I think we have a swap out video. So we're just undergoing a ship move, and while we wait, I am going to give you some Azores, not trivia, but esoteric knowledge. So the islands in the Azores were first known to the Europeans in the 14th century, and parts of them appear in what is known as the Catalan Atlas, which was created in 1375. Now, the Catalan Atlas is a medieval world map, also known as a mappamundi. And it was created in 1375, and it's often described as the most important map of the Middle Ages in the Catalan language. And, interestingly enough, it is the first known usage of the compass rose. Yeah, so pretty interesting. So if you ever have looked at a map and you saw a compass rose and you wonder where it came from the mappamundi of the Catalan Atlas. Now we make mapping products as well, and we also have roses and legends that we use. Those mappamundis were a little bit more flourishing than ours. We don't have mythical beasts on ours or dragons or anything. We can try and convince Shannon to put those on some of our mapping products, but I don't think that would fly. And so as we move, we see significantly more of these highly fragmented and faulted pillow basalts, and hanging out in the little pukas and the little holes, we see some shrimp. These are the ones with those characteristically really long antennae. And so we're in kind of a depth range. We started at a depth of about 1,930 meters below sea level, and we're going to try and come up to about 1,600 meters below sea level. And this is really targeting a depth range that we haven't seen on our previous dives. We haven't gotten into this range. We've kind of been above it and we've been below it, but we're interested in what the diversity and populations are and the speciation in this range. It's been a little bare compared to what we've seen previously, especially on those lower slopes when there wasn't a lot of hard rock. And now if we come up and we see more of this exposed hard rock, we're getting a larger percentage of sponges. Our ROV, this is a watch lead. The Scott is really determined to get to the top, and so just keep that in mind, and don't let me sidetrack us. Mm-hmm. I think so. Let me put my widgets on, and yeah, I would go. Let's shoot for waypoint 2, which kind of gets to that local high before it doglegs to the south and kind of goes up that ridge to the maximum high. Yeah. Yeah, waypoint 2 is a hard target. Yep. We can reevaluate that when Scott comes in and see what we see. Now, similar to where I live in Hawaii, the Azores has a series of what we call geohazards, or hazards from geological phenomenon. Those are things that you would expect. Earthquakes, volcanoes, tsunamis, and then also landslides and mudslides that are generated by them. And since about 1450, when the Europeans started keeping records, there have been 26 eruptions, both on land and underwater, that have been detected in the Azores. 4 of the islands of the Azores, Santa Maria, Guasoya, which I'm probably horribly pronouncing, I'm sorry, Flores, and Corvo, have no historical eruptive activity. The most recent volcanic activity was on Sao Jorge Island, which was a volcano-seismic crisis. So this was a swarm of earthquakes in March of just this year, and there was more than 2,000 magnitude 1.5 to about magnitude 3.5 earthquakes. We see these also very commonly in Hawaii as part of my job of monitoring seismic activity in Hawaii, and these are usually indicative of what we call dike intrusions. So this is magma moving in the crust, either laterally or up to the surface, and as that magma moves in the crust, it fractures and breaks rock and causes earthquakes and seismic activity. Now, at least in this case, in March, there was no subaerial eruption or detected submarine eruption, and this was under the island on the island's northwest rift zone. So a rift zone is a long, you can think of it as a conduit of magma that radiates out from the central source of volcanism. Now, in 1811, there was a submarine eruption, and it formed the island of Sabrina, but it later disappeared from erosion. So that's also very common in submarine eruptions. You might get a really voluminous eruption under the water, and that builds up one of these volcanic piles, this composite of maybe some pillow basalts, but also explosive products, pumice. It's probably very weakly consolidated. It's not very cohesive. It's not stuck together, and it'll break the surface of the water. But then over the next months to years, wave action and ocean currents will wipe it away, and it'll disappear again. Yeah, beautiful view on Stream 2 of just how steep. Wow, 67 degrees. That is a very steep ridge, and that's very indicative of what we see in kind of faulted areas, or areas that are also accumulated by talus. When we look at submarine volcanism, we tend to see slopes that are anywhere from 15 to 25 degrees, and that's based off of the viscosity of the magma that's erupted, and then also how quickly it cools. You can imagine lava that's erupted underwater cools very quickly, and it tends to stack up, whereas lava that's erupted in the air can kind of just be more runny, at least in this case with what we call basaltic magma that tends to be very fluid. On these talus slopes that we see, we tend to see slopes upwards of 35 to 45 degrees, and that's just kind of the natural angle that material settles at when it's accumulated as piles, as rubble. We call it the angle of repose. Now, on these faulted areas, we can get a whole range of angles. We can get shallow angles, very steep angles, depending on the faulting that's occurring. On these normal faults, we tend to have very steep angled walls. And so it almost looks like we have these ridges of harder rock, and then maybe these little chutes, these little valleys. You see one kind of on the right moving into center. That's probably where more sediment accumulates. And that's just a natural low in the topography that allows for accumulation of marine detritus and sediment. But I'm surprised on how superficially barren it is of kind of the macrofauna, the epifauna, the large scale, easily visibly identify seafloor communities. You know, we see occasional sponges outcropping here and there. And so intermixed with all these basaltic blocks, so a lot of this basalt is, you can see when it's kind of almost looks like it's freestanding on most of the free surfaces except the portion that's just touching the ground, that's almost always rubble. And then intermixed with that you see all these pervasive coral skeletons. We saw these on a lot of our other areas. Now it's unknown whether that'll actually be a good sign for possible coral communities above us, whether this is material that's fallen down as rubble or a lot of it was coral that was grown in place here. Something. We just put it in another move.
Yeah, so we actually did see this sea cucumber before, but we saw from a different angle. It was doing the same sort of thing, being lifted up from the surface at the anterior end. So this is another one of our deposit feeders. So far what we've been seeing today, we've seen, I think, 3 different species of sea cucumbers that feed on the sediments, and otherwise most of the slope has been dominated at least the size of stuff that we can see by sponges and bryozoans. And I was listening to some of the things that Ashton was saying there while I was at lunch, and it made me reflect on a couple of different things. So overall, the slope is quite depauperate compared to what we've been seeing, and that tells us that, you know, just looking at the topography doesn't tell us everything when we're making predictions about what might be a good place to dive for sessile suspension feeding communities. Currents are really important, being able to get the larva to a place, how stable the substrate is, all of that factors in. But the other thing that it tells me is how lucky we are when we do find really dense and diverse communities. And we have at least 3 or 4 dives of our first 9 encountered communities like that. And it tells you how precious they are and how careful we have to be about protecting some of these areas. And I guess the point I'm trying to make is not all seafloor is the same. We've just passed above the 1,800 meter mark. We descended through the water column. We landed on this ridge, Kai Ridge, at 1,921 meters. And as I say, we just passed 1,800 meters, so we've come up 121 meters in depth so far. There's one of the bryozoans that I was referring to a few moments ago. We've seen several of these fans actually today. Yeah, I'm not sure if that's a little encrusting sponge up here or if it's chitin. We're going to have a zoom on this. Yeah, good eye rolling. That looks like a chitin. So this is a kind of mollusk that related to the snails, has its shell divided into 8 plates, and you can see those plates quite nicely. They're called valves, and then it's got a muscular girdle around that helps secure it or suction it to the rock, and this scrapes the surface of the rock, and down here presumably would be feeding on bacteria and the various organics that have settled onto the bottom. Very common in wave-swept rocky shores, so you can see lots of chitin yourself if you just go down to the shoreline, if you have a rocky shoreline nearby. Yeah, let's have a look at that. Thank you. I suspect this is going to be a sponge. I'm trying to replay already what we've seen today and not mix it up with what we've seen in past days, but I'm not sure that we've seen a yellow sponge today. That may be in the genus Hertwiggia. We've been seeing a lot of Hertwiggia sponges this week in different areas. I know there's at least 2 color morphs, a yellow one and a white one. There's kind of a globular sponge just right of center, but then right above that, to the right and above that is something else. I'm not sure if it's a sea cucumber. It's between the two sponges that are in a line there. Yeah, it's another one of these sea cucumbers that's kind of wafting up at the anterior end, the head end. So this is the third or fourth of these sea cucumbers I've seen. All of them are doing this behavior, and I thought at first it was just the ROV prop wash, basically. And now I'm less convinced of that. And I do know that sea cucumbers will get into kind of like mating swarms. They will aggregate, and several individuals will raise up off the surface like that, and they will release their gametes into the water column at the same time. And the idea there is that because the deep sea is such a big place and these things are so scattered, there's some cue that they're using to all release their sperm and eggs into the water column at the same time, and that increases the chance of successful fertilization of the egg. So I don't see any clouds coming from this sea cucumber, so I don't know that that's what's happening, but I'll just offer that as a hypothesis. And then we have, it looks like, two isopods crawling over the bottom, one above, and there was another one just below off screen. We've seen several isopods today, which is kind of interesting because we don't often see that many, and we've only seen them a couple of times on this expedition so far. So kind of neat to have the opportunity to see these a little bit more closely. They're very common down here. A small jelly drifting upside down, drifting from the right to the left. I say upside down, but maybe this is the way this jelly likes to swim. But you can see that marginal bell, that umbellar space, uses muscular contractions to sort of jet propel itself forward. Hanging down in the center of that space, excuse me, is a little tube that would bear the mouth. And I think those sort of brownish pockets that you're seeing inside are the edges of the radial canal of the stomach inside that bell space, or within the tissue that is enclosing that bell space. Alan Collins from the Smithsonian says that looks like the genus Egenura, a pale version. Nice frilly glass sponge here, possibly the family Fariidae, excuse me. Goodness, Alan Collins giving me a name here. I got to think about how to pronounce this. That may be a possible identification to the genus level of that jelly that just went by. He doesn't really recognize it, says it's a good collection target, but it could be Sigiwedelia. Trying to ascertain which of the many different kinds of sponges we're seeing. So I'm not 100% convinced myself. But if you could let me know as we're going when you think you're in a good position to collect, and if I see any, I'll let you know. It might be the one on the left. I don't know if, yeah, far left. It's possible at all three of these. No, your other left. Yeah. Oh, you're. My bad. My bad. I see. I see what's happening. The ROV moves a little bit more slowly than my head, so I apologize. Yeah, I suspect this is a target. Yeah, I have confirmation from shore. It's these type. They're kind of brown and spiny on the outside, and when you look through that opening, they are very white on the inside. Excellent view looking in this sponge. Again, you see some of this architecture, and most of the, well, the architecture, the framework that supports this sponge is a combination of these spicules, which are made of silica, and come in a whole variety of shapes and sizes. Mostly what you're seeing are these spine-like or needle-like ones, but there may be a whole diversity of other shaped ones inside, and it's actually the shape and I guess abundance of each of those types of spicules that is largely used in sponge taxonomy, naming, and identification. But then they also create a, I guess it's a protein called spongin that's a fibrous matrix that allows for the bulk of the sponge to exist, and then there would be various mucus inside there and water. So the type of true bath sponges that you purchase would be mostly the spongin fiber, and they wouldn't have the spicules. There's been no shortage of these, so I think that's a good plan. Yep, and there have been for much of the slope. Yep. Again, for viewers on shore, if you're watching on stream 1, switch over to camera 2 and get a sense of this really sheer, tall slope wall that we're climbing. Yeah, it's pretty impressive. This would not be easy to scale. Sometimes in the maps that we produce, in order to highlight features, the mappers turn on an exaggeration. And so I think on the dive plans we send out, we do what's called a 3 times exaggeration. So the height would be 3 times the horizontal distance. And so it may appear that bottom is going to be steeper than it actually is when we get down there, and that is not the case today. This is as advertised in the maps. Yeah, and I'm not sure if you heard the video on the OKX call, but he was noting, it's so steep that you would expect when skeletons, when organisms do die, they're going to tumble down slope. And so the question is, all this coral rubble we're seeing, is this tumbling from that crest above us, or is it more a reflection of something that was growing on this slope thousands of years ago and is just still sitting here in place and is basically a subfossil? That would be more challenging to answer and would require analysis of these skeletons themselves. And we did suction up a few bits of those before, so it wouldn't be impossible to address that question. I thought we were going for the red one too. It's a little Anthomastus coral. I think that's only the second coral that I've seen all morning. And then I'm not sure how our video engineer got to see this, but there was another octocoral just at the top of the screen. We'll look at that next. Yeah, so this is, we sometimes call this a little bit of a mushroom coral. You can't see it, but below that red ball there's a stalk that would be attached to this this dead coral skeleton that you see. And then it's got the bulbous section, and then you see the feeding polyps that are extended from it. And this is an octocoral. You see it's got these 8 tentacles, each with their little side branches called pinnules. So those are the hallmarks of the octocorals. And those polyps can retract. They can compress all the way into the tube. The upper polyp, you see that there's this pink line that goes all the way down from where the tentacles are and just kind of disappears into the red ball. That's its pharynx. That's basically its throat. And the surrounding area around that pharynx would be the equivalent of our stomach, its gastrovascular space. So just below the level of that ball, that tube would end and then open up into that space. The other thing is, oh yeah, I see what you mean. Yeah, so immediately right of this coral that we're looking at is an old bivalve shell. I'm not sure if it's file clams, something like that, but it's completely crusted over, and you can see the ridges running along it there. Yeah, nice view. So this one's going to be kind of white. Doesn't really have color associated with it. It'll come in view in just a moment. I wonder if it retracted the polyps. I noticed there. Oh, it is there. It's at the bottom of the screen right now. Sorry. It's like to the right of the hermit crab. Now it's almost in the dead center. It's a little bit above the lasers now. There we go. Just coming into view, and again you can see 8 tentacles pinnate. I'm only seeing 1 polyp, but when we were zooming in on that other one, I think I saw at least 3, so that means the others have retracted themselves. Actually, you see just above and left, there's 2 what looks like little thumbs sticking out, and those are the polyps, what they look like when they're completely retracted, so that's kind of nice. This is something that we call a stoloniferous octocoral because it grows as a little ribbon-like mat instead of producing an upright skeleton. It just overgrows other surfaces. So very cool. Thank you. And this must be a tunicate, I think. Well, we're getting all kinds of unusual things today. Yeah, okay, so this one I can actually see. This is a compound tunicate. Beautiful, beautiful. So this is a filter-feeding organism. It's in the phylum Chordata, so very closely related to the vertebrates. And I'm seeing it's a ring of individual zooids. And on the outside, you can see these small openings. Those would be incurrent. And then you can beautifully see the pharynx with its filtration basket. They create these baskets that they coat in mucus, and there are cells in there that generate a current flow. So they're pulling in water in those outer holes. It passes through the pharynx that is lined with mucus, and so organic particles get caught up in the mucus. And then the water then passes out that central large hole instead of being funneled into the gut. But the mucus with all the particles is carried along to the mouth that's on the inside there. That would be a little bit harder to see. And then processed like normal. It otherwise has a gut and an intestine. And the anus to those things also opens up inside that atrial space, but nearer that large opening so that the outflow water carries away the waste products. That was great. I've never seen one like that in the deep sea, so beautiful image. Gorgeous. Can we have a look, pilot, the top left? I'm seeing a sponge, but it looks like there's something on it. Thank you. Thank you. I can't tell if this is like a crab or something that's on the sponge, or it's a different sponge or tunicate sitting behind the sponge. And there's another one of the tunicates that we just imaged, kind of like at 5 o'clock, just in front of that sponge, about 2 sponge distances away. Right there. Oh, I see. There's something coating the rock. Oh, and there's another one of the 2 more of those tunicates up there. I'm going to guess that the sort of purplish color may be an encrusting sponge on the rock. It's hard to tell. But the sponge in the foreground is interesting. Also very different looking. With all these silica filaments sticking out from it. I don't know if what I'm looking at is a very young version of something that I would recognize if it was much bigger, or if this is something completely different. And you can just see the different texture of whatever that is that's growing above the sponge. Again, I don't know if it's tunicate or another sponge. Kind of looks like a different kind of sponge based on those large holes and lobes at the top. Yeah. This is an interesting and mysterious place. Thank you. Pilot, can we, if we zoom out, there's a rock that has 2 of these yellowish tunicates, or 3 of them on it, and I'm wondering, Ashton, maybe you have an opinion if any of these rocks are loose. Not the one back there, it's in the foreground, but that's what I'm looking at, but it's closer to you. Yeah, there you go. Just to the lower right of that sponge. Did that rock look like it's loose, or you think these are all in place, you know, like encrusted in place?
Which one are you pointing to?
This one right there.
Oh, I think that's...
Just to the right going off the screen now, you see there's 3 of those yellow globule things.
You could nudge it, but I'm guessing it's going to be pretty...
That's what it's looking like to me.
Together, yeah.
Yep. Okay. Thank you, pilot. We're done here. I appreciate that input, Ashton.
I try. Thanks for including me.
All the time.
I was sitting here reading my Wikipedia article about tunicates, so I could yeah,
join in the conversation.
Yeah.
Well, here you can tell us what you see here.
I see a bunch of yellow chambers.
Yeah, each one is a zooid, so each one is basically an individual member of the colony, and it does kind of look like they're bound together in a common matrix that's growing over the rock.
And then what are those little, these little yellow fringes at the bottom that don't seem to be part of the individual tunicate, but maybe part of the larger matrix?
Yeah,
are those?
That's a good question, because I don't see them so much on the top. Yeah. I'm not sure. If I had to guess, I would guess they're either forams, except there's so many in a row. And I noticed that there's a lot of like networking going up here. They seem to be interconnected, so I suppose it's possible that it's a hydrozoan that also grows as a map. And there's yet another isopod down there below in the rock. We've just seen so many isopods today. We've come up almost 25 meters in this slope since you went to lunch. Let's have a zoom coming in top center. Thank you. And that looks like a pom-pom anemone. And whenever I see these, for some reason, the genus name immediately leaves my brain. One of the things I remember about these anemones that's so unusual is they have deciduous tentacles, meaning they can shed their tentacles when they're disturbed. There's a coral down there on the rock. Liponema, there we go. That's the genus for the pom-pom anemones. So I don't know for certain that's what this is, but that's what it reminded me is. Sure looks like it. So somewhere down in there among those tentacles would be the mouth. So anemones are free-living polyps. If you're more familiar with jellyfish, think of a jellyfish, attach it to a substrate, and you basically have an anemone. There's some finer detail changes when you look at the internal anatomy, but that's essentially what you have. So tentacles are presumably packed with various kinds of cnidocytes, which are specialized cells that contain organelles that fire little threads. Some of those threads are sticky, but some are the well-known stinging threads that have various toxins associated with them to paralyze their prey. So getting an agreement that this is probably Liponema. There's a couple more of those tunicates, those yellow tunicates. Kind of interesting. Haven't ever seen one in my life, and now I've seen about 10 in the last 10 minutes. Tells you something about the patchiness of distribution of organisms in the deep sea. Just making some notes there in the chat room to some of the scientists. So this is another glass sponge, vase sponge, and there's an awful lot going on inside this sponge. It almost looks like there's a coral growing on the inside there. I'm not sure that the sponge is in great condition. It seems to be being overgrown maybe by a network of hydrozoans, and I see a worm tube on the side there with maybe a worm sticking out the front. And just so you know, that's looking down the side is great. It's not a critical thing. If it's going to slow you down, then yeah. Thank you. Love it. Joanna Javier is joining us now in the chat room. She'll be your biology lead starting next week, and she's a sponge biologist, and so you'll learn a lot more about sponges, but she says this looks kind of like an Asconema. Oh, you're passing over Chrysogorgia. That's a good sign. That'll be the first Chrysogorgia octocrawl of the day. Huh. That's a very good sign. Not sure how I missed that going in towards that sponge.
This looks like a large flake. That outer crust that's flaked off, and you can see the shadow underneath it. That flake with all these holes we're seeing makes me feel like there's probably a lot of subsurface or fluid circulation.
Mm-hmm. It's a crinoid hanging on just under there. And when we zoom back out, can you just look again to the left, please? I have to remember, it's never easy to ask to look right or left because the whole vehicle has to turn. Okay, I think I thought I saw coral. I can now see it's a track on the seafloor, so we're good there. Thank you. Joanna says that on EX2206, sorry, Voyage to the Ridge 3, Back to the Ridge, or was it Return to the Ridge? What did you prefer?
Return to the Ridge.
Return to the Ridge. Conversely, people will learn a lot less about corals, but a lot more about sponges, which I think is a good trade-off. Another Liponema anemone there on the bottom. Here's another one of these pipes that we saw earlier that looked like a dead encrusted sponge. It does have at least one tunicate growing on its side. Maybe two tunicates growing on the side. Interesting. So two tunicates, the yellow things that are on the side, those are colonial compound tunicates. And then there's some sponge growing on the top. And the way I'm interpreting what I'm looking at is that the main structure, I guess perhaps it's a dead coral, but I thought it was a dead sponge. But that there are subsequently sponge larvae have settled on the top and are growing. There's at least two of them. And then we're going to look down here, and now you're seeing the tunicates that have settled on here as well. Yeah, that looks like an old encrusted sponge skeleton. That tube that's extending off to the right, that's probably a worm tube. The worm is retracted on the inside of it. Great stuff. All that sort of messy, hairy, wiry stuff that you saw at the base there, that was also sponge spicules that would be used to attach to the base. There's another crinoid on the surface on the far right. Not sure what that is. More coral rubble in this little chute-like feature. There's another black coral. Oh no, sorry, it's the arm of another crinoid, this sort of brownish red crinoid on the rock. We've only seen one black coral whip today. And then we saw the one stoloniferous octocoral, one anthomastus, and then we just shortly ago passed over one chrysogorgia, which is just remarkable given the thousands and thousands we've been seeing on these earlier dives. So there's a nice view of a crinoid. It's another kind of echinoderm. We saw some sea stars earlier, so this is a cousin of the sea stars, but they feed with their mouth oriented up away from the bottom, and they raise their arms into the water in order to pluck food out of the water column. Let's see. Can we look to the left? I'm noticing something brown. I don't even know what I'm looking at. It's almost... What do you think?
I think it's the shales.
Oh, yes, you're right. Okay, thank you. We can, yep, let it go. Thank you, Roland. There's various of the thread-legged shrimp that are crawling over the bottom. This kind of looks pillowy, Ashton, to me.
Yeah, I think that's a large fragment of a pillow that's been kind of sheared off by this normal faulting process.
Mm-hmm. At least I could see its structure there.
Yeah. You're getting a good eye, Scott.
Thank you. Another one of the Liponema anemones up here. There we are. And one of the ways we can tell this type of anemone, Liponema, is that, it's really hard to see where the mouth is. There's just so many tentacles, and they're probably compressing the oral disc somewhat. Pentametrocrinus. Thank you, Asako, through Tina, or thank you, Tina, through Asako is the name for that crinoid with the 5 arms, that reddish-brown one. This is another dead sponge with a live crinoid on the side of it and a living sponge right there, just coming into view, that white one on the top. So remember, in order for all these animals to get around, there is a larval stage that swims through the water column, and it can settle on various kind of substrates, and so in this case that larva must have settled on the top of the sponge. Was now going to make a go of it on the sponge. So even though the sponge itself is dead, its skeleton is providing habitat for a long period of time. Boy, 2 or 3 more of those yellow tunicates. And this is another one of these tubular constructions. I'm trying to decide if it's a sponge or, notice the base looks really wide there. So maybe it's a coral skeleton. But they all have the same structure, and they're capped by a live sponge. It's very interesting. Yeah, let's go for that. I'm just typing in the chat room right now. So this looks to me like an octocoral, likely in the genus Victigorgia. Yeah, very interesting. So again, we've seen now a few octocorals. The polyps have 8 tentacles. They produce these skeletons in a variety of different ways. And there aren't too many species in the deep sea that have this purple color. And the two choices, to my mind, would be Victorgorgia or one of the mat-like growing ones, Trachythela or Clavularia. That's a gorgeous view. So in the center of each of those rings of tentacles, there's a mouth that leads down that tube that you see on the inside. Oh, actually, the top center one is fantastic. You see the tube comes down. It's purple. That's the pharynx. And then you see what look like 5 frilly lines leading from that base of that pharynx. Those are called the edges of the mesentery and mesenterial filaments. And so that junction is where the gastrovascular cavity starts and kind of opens up. And on the edge of those filaments are cells that secrete enzyme and also cells that have more nematocysts. So if they do ingest, let's say, a shrimp and it's still alive, they can still be stinging on the inside of the body to subdue it. And the other thing, you can kind of see through the tissue of the wall, there are these little lines crisscrossing back and forth. And those are calcium carbonate structures called sclerites. And they provide some support to the polyp, and for a taxonomist, they're used for describing a species and identifying a species. Very nice. That was great lighting.
Notice these are in different groupings down the stalk.
Yeah, so, yeah, that's a really interesting observation Roland's just made, that the polyps are not evenly distributed along the stalk. It's almost like there are little swellings here and there. And as I said, there's 2 possibilities here, that it's a Victorgorgia or that it's this mat-like growing or stoloniferous-like, ribbon-like growing one. And maybe it's the latter, given the way that there's an irregularity to the way the polyps are rising. So what you're looking at crawling over the surface, that orange thing, that is a scale worm. And I'm kind of interested in those 3 white dots on its back. I think that might be a parasite. It's possible, I suppose, that they could carry their eggs. Scale worms have a what they're called elytra. They're scales that arise from their paddle-like legs and extend over the back or the dorsal margin. Oh, there. Actually, sorry, I'll come back to the worm in a second. That's a crinoid. I can see now the base. So the quote-unquote skeleton for this coral is actually a stalked crinoid. So this is not Victorgorgia. This is absolutely a stoloniferous octocoral, grows as a mat, and it's just taking advantage of the crinoid skeleton and growing over it. Great view. So yeah, I don't know if that scale worm is storing eggs up underneath its elytra, or if there's a parasite there. I'll see if anybody on shore has seen anything like that and can give us some indication. It's another neomorphaster, excuse me. No, we viewed this earlier downslope, and Chris Ma came in the chat room to identify it. Neomorphaster sea star, the one with the very long arms there. Oh, I'm hearing there's a coral coming up. Oh, nice, yeah, Chrysogorgia. So I thought we passed over one of these before, and we kind of missed it on close-up. So this will be good to see. So if you've been with us on previous dives, you've heard us talk about the Chrysogorgiidae a lot. It is a very common family of octocorals in the deep sea. There you go. They have this very characteristic growth morphology of that main stem. At certain intervals will angle in a kind of a spiral fashion, but instead of a smooth spiral, it's very angular sort of thing. And every once in a while, at the change of angle, it puts out a branch. And so it gets this very regular geometric appearance, and it's one of the ways that they use to put the different genera into groups. And you can see that the polyps are really small and delicate and almost perched on top of the skeleton. And it may be hard to tell, but all of those polyps are interconnected. There is a very thin layer of tissue on that skeleton, and there's an amphipod in the center. Excellent, thank you. I'm sorry, Joanna, I missed. Oh, she must have meant the barrel sponge in front of the Chrysogorgia. Sorry about that. Joanna, we've seen a lot of them, although we were also seeing a lot of the reddish, what we call the pineapple tunicate, and I haven't seen those in a long time. We tried to collect one. We weren't successful. And this is a thing, you know, one of the well-known patterns in the deep sea for biological communities is that there's depth zonation. And those zones can be several hundred meters wide, but there are certain places you get where you get a turnover in the species that you see. So it's a challenge to say, well, I'll wait till I get a little bit more upslope and I'll collect something, because you may be transitioning from one depth zone to another, and so you lose those species. Yeah, on the left side there? Yep. I think you are correct. And okay, and noting a different kind of a coral, might have been a bryozoan, was growing off the side of that rock. Just passing 1,745 meters. It's Chrysogorgia again, I believe. Maybe the same one that we just imaged. And yes, it is. It looks like it even has an amphipod in the center again. The star behind it, I don't know why I'm blind and not seeing that. Oh, there we go. That looks a little bit like the neomorphaster we saw earlier, but maybe not. This might be a different one. That might be a different one. Yeah. So Chris Moss says this is Henricia. This is, I want to use the name Henricia for any of these sea stars that look like they're very elongated legs and thin legs, or arms, I suppose I should say. Sorry, not legs. Arms, arm rays. So that's good.
What distinguishes an arm from a leg? Because you used the word arm, but then you corrected you used the word leg and then corrected yourself for arm. So,
I mean, they're both generic terms, neither of which I know have a specific meaning in an invertebrate like that. It's a ray, right? And so for vertebrates, for tetrapods, arms would be your forelimbs, and legs would be your hind limbs. You know, they're both kinds of appendages, but this is a cup coral. First cup coral I've seen that's live today. We've seen many of them dead and encrusted in this, whatever the black incrustation is. And so this is a really nice view. It's a scleractinian coral, stony coral related to the ones that are building the big reefs in shallow water. But it's solitary, so there's only one polyp associated with this, and it won't ever produce a second polyp. It won't become colonial. And you can see the transparent tentacles are studded with these little dots. Those would all be the nematocysts, the stinging cells, in little bundles. You're not seeing a cell itself, but you're seeing an aggregation of them. The mouth is in the center. And then those white raised ridges that you're seeing are called septa, and they more or less subdivide the inside of the polyp. And I'm not sure if it's got a functional basis for it, or if it's, or what, but all these scleractinians have those ridges. There are multiple numbers of ridges, and the size of the ridges and whether they extend on the outside of the cup are also used in the identification and taxonomy. Yeah, so we've been seeing this sponge several times today, this beautiful blue color.
Now, do you think that blue would leach out if you try to preserve that specimen like you did the coral?
That would be my best guess. My experience with color in sponges is that the pigment is embedded in the tissue and not the hard parts, and so that usually makes it mobile, and so when you put it in ethanol, you end up with a colored ethanol. Now, sometimes there's a reaction, possibly with the oxygen, that turns that pigment to a different color, black typically. I don't know if that's the case with these blue pigments. So there's another one of these bivalves, I think.
Well, look at the
dead shell.
Well, I was saying you can see that real deep kind of hole behind you in that cavern, and then it's been partially concreted over by the coral that's fallen over it and kind of bridged it. Yeah.
It's like a little bridge.
Yeah.
Right here. Cup coral bridge. Yeah, I see Joanna's asking, is this subfossilized coral rubble? That's something that I was proposing earlier, and Ashton was saying it's a little hard to tell just from this view.
Yeah, you know, on the one piece we picked up earlier, that was heavily remineralized and no real presence of fossils in it. This looks like it is, but, you know, we can collect a sample and see how concreted it is. But at least, you know, on what we've kind of pushed around with the manipulator, it seems to be pretty individual fragments, which means to me it's probably not too old. But this also gets into one of those dangerous areas of me trying to talk about sedimentary processes.
So I think that's an anemone that you're seeing, or a corallimorph even, on the left side, that large polyp. It's a sponge in the center, and then the white branching thing underneath that is a bryozoan. I think we have Megan McCullough in the chat room, and she's a bryozoan specialist, and she's been hearing about the bryozoans but hasn't been able to tune in. So this is great that she'll be able to get a nice view of this. There's been quite a lot of bryozoans today. Yeah, I kind of think this is an anemone, but I'm not 100% certain. Sea anemone. And the other choice would be Corallimorpharian, but it looks more anemone-like to me. That raised structure in the center of that disc among the tentacles, that's the mouth. There's only one opening into the body of an anemone. All these polyps, there's only one opening. So pulls food into that digestive space, and because there's only one opening and there's no intestine, it's just basically one room, if you will. All the digestion takes place there, so they have to basically digest their meal and then get rid of the undigested comments back through that same opening before they take on their next meal. Unlike organisms that have a flow-through gut with a long intestine, they have different specialized areas of the gut so they can continually be putting in food, and it would be at different stages of digestion as it passes along the alimentary canal or the digestive canal. Sorry, I'm typing. I'm trying to communicate with shore and see what's going on here. So we're seeing, yeah, this is an interesting cup coral. This one looks quite different to me. It might just be the angle. And then another stoloniferous octocoral of some type. That's the stuff that's kind of at the right side upper, and the polyps are being blown upward by this current. And then maybe a hydrocoral. That's the thick branched structure above these other ones. I can't tell if it's still alive. I'm not even sure that that cup coral is alive, but possibly. Michelle Sharer is asking, are the tunicates and bryozoans here indicative of exposure to high currents along a steep wall? In fact, I think it's the opposite. I think that we have relatively low currents, and that's why we're seeing so many sponges and bryozoans and tunicates instead of a lot of the larger corals. But that's just a guess on my part. Yes, I'm just trying to communicate a message to those of you on shore, especially those of you who are interested in the sponges. I'm not familiar enough with the sponges, and especially some of the names, and the delay that your comments come in the chat and my ability to read them means it's not helpful to say that sponge there or this one. I need more descriptive identification. Now I know what the pilots are like when I'm back here asking, look at that one. No, the hemicorallium, not the metallogorgia. So, yeah. Okay, more typing in the chat room. So I think this is another little glass sponge. I don't know if that's family Furiidae. It's not as frilly as I would expect them to be in that family. Looks more like a series of tubes. But we do have Joanna Javier on the line, and she would know much better than me what that is. Actually, I'm not sure if you had seen in the chat room, there's an interesting discussion about these potential subfossils, and Les Watling was noting during a 2005 cruise to the Mid-Atlantic Seamounts, there were a group of researchers, Jess Adkins and Laura Robinson, who were collecting lots of these subfossils and doing some dating and aging. And I'd referred to this earlier in the dive, but basically that they could do some reconstruction of paleoceanography, and the cup corals that they were targeting, they were able to date on this feature, I think it was about 25,000 years ago, and so interpret what the climate conditions were like at that time in the seamount. And one of the ways they did it is not just by looking at the rubble, but because the cup corals were still attached to the rock in place, you could very clearly indicate, what depth strata they were in and so on. So that was a Furiidae that we just passed. Joanna suggested genus Furia. Here's another one of these very lacy Chrysogorgia colonies just coming into view on the lower right. We've passed now 4 or 5 of those. There's actually 2 of them. Again, it's giving me this sign that up above, although it's still pretty far up above, we may get into the zone, especially if the dominant current is coming from the west, where it passes over the top of the crest and provides more opportunities for feeding by the larger suspension feeders. Not that I don't enjoy the sponges are beautiful. Oh, so that's interesting. So a lot of these globular sponges that we're seeing, we had some information early on that they might be a target of collection, and Joanna's just said that it looks like, based on the photos that have been forwarded to her, it may be a new species that she has in the lab right now that they're describing. So indeed, it is an unknown species, but they have a sample and they're putting a name to it. Oh, can we look, just coming into view upper right, I think there might be a live clam finally behind those sponges. I wonder if Jean-Marc, I see his name in the chat room. You'll have to let us know if this is a file clam or if this is something else. But no, it's a tunicate. You can see that it's really gelatinous as we zoom in. So this is another colonial chordate. We've been seeing these yellow golden ones that are compound tunicates. That's a different species of the same. In fact, there's this globular yellow sponge on the left, and then just to its immediate right, that's yet another tunicate. Again, it's quite gelatinous. There's a couple of them here. And we managed to collect, I think, at least 2 of these in the last week, which is interesting. Maybe I've said this a couple of times. I don't recall very often being able to collect colonial tunicates on these expeditions, so those represent really good collections. Thank you, pilot. She's referring to the globular sponges that we were talking about, the Thynia. Yeah, so some more input from Joanna. She said, we recently found 3 new Thynia species. So again, that's the globular sponge that's kind of got the brownish coating on the outside. 3 new species from the Azores region, all collected considerably deep, below 1,500 meters. So we're coming into view can we get a look at the sponge on the lower left? In the center is another one of the Chrysogorgia. I've seen several of those, but I don't recognize the texture of this sponge. This is kind of interesting, so Joanna, stand by for this one. And she says that this one in particular looks like it's their Athenia species 2, new species 2. So stand by for the publication. We'll actually have a real name for it. Cool. Look at that. As I said earlier, these glass sponges have so many interesting architectures and frameworks. This one's got a barnacle growing on the back, a couple of gooseneck barnacles. Thank you. There's an anthotheelid octocoral just below there, the cream-colored body with the polyps arising from it. Standing by for Joanna to give me some input on this sponge. I'm not sure what that bright yellow-red thing was. What else do we see here? It looks like a bunch of hydrozoans. That's the thin beigeish branches that you see. There's a whole bunch of them overgrowing. The bulk of the structure is a large dead sponge, and then the small white globules are other sponges growing on it, and then there's a Faria sponge growing also to the left. But this one, not quite sure. To the right. Oh, nice. Good work, Roland. Yeah, so those are corallimorphs for sure. So you see there's three, I think, two polyps right there, and then maybe one more just off the screen there. And you see how transparent they are. And I keep using the word diaphanous. I should look up what that actually means. But to me, it means delicate and see-through and kind of, I picture this soft sheet waving in the wind, you know. Yeah, did I get it? All right, good. So a corallomorph is another order of the hexacoralia, so they're related to the black corals, the sea anemones, the stony corals, and the zoanthids. Very cool. Joanna says thank you, yeah. Says she's never actually seen this sponge live. She thinks it's possibly in the genus Hexactinella. I don't recognize it either. Are we in a position to collect, pilot? I realize we're on this cliff and everything's moving. Just let me know. The whole structure that it's on is a dead sponge, and if you could just grab that dead sponge, we would get a bunch of things there. So you tell me about the comfort of your position. Yep. Yep. Understood. I think it's worth it. There seems to be quite a lot of excitement from multiple scientists on shore, so I think it's worth it. We haven't made any collections, so let's give it a shot. I totally understand the potential problems involved. Yep. Copy that. Yep. I got the risk. Understood. And let me tell you that the main target is that very interesting textured white sponge. So if you can grab the structure below it, the whole structure should come off the wall. And by below, I mean to its right. Don't sloughy. Yeah, copy. No, I can see the problem. Yeah. So the one that's floating there was the primary target. I don't know how easy it is to grab a floating sponge. The suction would be fine with us. It'll break up a bit, but that's okay. Yep. Oh, I see. Got it. It's really delicate. I don't know how to grab it. All right, we all knew that was high-risk pilot, so please don't worry about it. We knew we were taking a chance. So we're hearing from Ken Sulak, who's just joined the chat room. He's the now retired USGS biologist who's expert in all things deep-sea fish. And he says the fishes today are relatively abundant and diverse, and this suggests a depth horizon and environmental context rich in diverse foods for diverse fishes. So that's interesting, especially given what we were talking about food with respect to the suspension feeding community. And he did note that that was a snailfish that we saw earlier, a small black-headed snailfish, Paraliparis, and is echoing some of the comments I made about the eggs. He said that the family is unusual and that they lay clusters of relatively large benthic eggs. Thank you, Ken. Oh, Joanna was also saying, you'll be pleased to hear this, that don't worry about the sponge bits that fall to the seafloor because they will probably reattach and continue after. Sponges have this amazing ability to repurpose. The basic vertebrates textbooks and so on is that they don't have true tissues. So unlike us, where we have stem cells that then grow and take on a certain fate. So, for example, a cell becomes a nerve or becomes a muscle or some epithelial cell on an organ. It can't then change later on to one of those other tissue types. Not so in a sponge.
Yeah, Nav, you want to do another 5 meters?
The sponge may be able to repurpose some of the cells to get the attachment point, grow new silica spicules, and continue going.
Sirius is starting to see some relief up ahead, so maybe we'll get lucky coming up, find something.
Did you guys get a mark of the depth where we were at when we sampled it?
1722.
Nav, did you mark that sample?
You want me to mark it now?
Yeah.
Yeah, get something.
Next time, just do it when he plucks it.
Will do.
Ken also notes that we were looking at this, the cusk eel, you may remember it was pretty dark. There was some initial debate about what it was. And he says that's stand by. This looks like yet another tunicate. What is going on with the tunicates here?
Coming down. Stand by.
Is it? And Joanna can tell me if that's actually a sponge, but it looks kind of tunicate-like.
You haven't seen a... Oh, did it really?
It's all right. That's the risk.
Yep. All right, I'm going to take the craft, turn it off, Sean.
Yeah, copy that. You can get settled for a... Okay. Is there anything left? We can look and see if there's anything left before we turn it off.
Sitting on top of the rock box.
I have permission to move H2 real quick.
Go for it, Sean. You can take more of this rolling.
Every time I zoom in, I think this is a sponge and not a tunicate, but from a distance, it looks like a tunicate.
Do you see anything in the JAWS video?
I think the video might be looking at a different camera. This is not focused. There we go. Thank you.
Sorry, I was asking a lot from video right there. Watching
it.
I
no, I understand.
Him.
Totally understand.
Yeah, Pied, if you're settled, what you could do is rotate the jaw hair towards Mini Zeus once you're settled, though.
Yep.
So, no rush.
Of the craft, yeah, on the starboard side. Yeah, it's sitting right there.
Yeah, but we won't reach for that.
No.
So Joanna does confirm this is indeed a sponge that we're seeing. And I think you can see that from looking through the largest openings, just the sort of network, and what look like a whole bunch of connecting branches and passageways are probably the canals where some of the sponge cells are. Pilot, if you're still in position, can you look down below? There's something a little spiny. If you have to move upslope, that's okay.
Down below?
Yeah. Oh, there it is. It just came into view. It's a little bit right of center. It's on the dark rock face. It looks like a little spiny ball or something, or fan.
Come in partial roll.
Just to the right of center, and it's white.
I can
See it on the rock face?
corner.
Yeah, now it's in the upper right.
Upper right.
Yep,
Gotcha.
right in there. Not
to
sure if
rotate
this is
over
another
side by side.
bryozoan or a little hydroid or carnivorous sponge. Could be any of those things.
I'm going to go with hydrozoan.
Certainly very small. Oh, it's another little bryozoan. There you go. So, Megan, if you're still here, it's bryozoan sponge day. Good stuff.
Maxim. A piece of sponges.
You can see, you know, you can pretty clearly see where each of the zooids is. It's kind of zigzagging back and forth. Thank you for that, Pilot. Yeah, so some more from Ken Sulak on the fish. The cusk eel is in a subfamily Benthic. It's viviparous. It's a rock-hugging megapredator. Another fish with an unusual reproduction mode in the deep sea. It's a live bearer. And it has a very enormous gape when the mouth is opened. So it's a swallowing inhaler of large prey. And I think somebody was asking what do those fish feed on. It's another bryozoan, a couple of bryozoan colonies down there.
It's another one of this.
Oh wow! It seems I got Bathygatus right. That dark grenadier that we're all wondering about. It's a special subfamily of its own. It's a hoverer drifter that descends only with the pelvic feelers that it uses to detect its prey. It's not a sediment feeder, but a swallower of small near-bottom nectins, such as amphipods, isopods. Several more sponges here. Different species. We've got this sort of plate-like sponge, a couple of them. Then this whole series of small tubular-like sponges. And I collected some of these off of a rock, one of the rocks we sampled. They were considerably smaller, but they were little bulbous structures at the base with a tiny little cone that erupted above them. Not sure if this is the same thing we just tried to collect a little bit earlier. We're just seeing it on a different face.
Coming back around. Full screen.
Joanna notes for many of these encrusting sponges that we're looking at, it's really hard to identify because the majority of species were described from dredged material, so a rock dredge or a net has been dragged over the bottom, and the material comes up and gets preserved, and so then the taxonomists are looking at the preserved material, so they don't know what they actually look like when they're alive. Maybe a very good condition, very different condition when you see it
Next
live in situ, inflated, color,
so we can check the jar
not torn up and broken.
See if there's anything in. If not, we'll just.
And I can attest to that by
turn
seeing
them
some
off.
of the sponges that we've collected, and after we preserve them, they change color. There may be some degree of shrinkage in some of them. Not the ones that have a solid skeleton, but and there's a very different way that
Looks
we're
like
collecting
we're
things
starting to
quite
get
delicately.
Yep.
And that wouldn't be the case when you have an indiscriminate dredge sort of going over the bottom where you're picking up rocks, coral skeletons, sediment, and various organisms. Everything gets kind of jumbled together like the dryer with rocks. Another beautiful glass sponge here, and the same stoloniferous or cornularid octocoral down there below. Nice. I don't get tired of looking at the intricate design of the silica skeleton. And this one, it's forming a series of tubes, large tubes, which obviously water is going to move through, but the action is taking place inside the walls of all of those tubes. Hertwiggia falcifera. So this is interesting. I mentioned it earlier. Oh, there's another one of those pineapple tunicates, the first one I've seen in quite a while. The sort of red structure, red-brown. So she identifies that as Hertwiggia falcifera, and it's the white morph, and earlier on we were looking at a yellow one that I thought might be the same thing in the yellow morph. So it's kind of interesting that they can grow in these two very distinct colors.
Don't know if I got a wide enough lens for that. I'm trying to back off.
There's a nice one. Yeah, I've seen things like this before, at least in the Pacific. I don't recall if I've seen these on Atlantic seamounts and so on. Megan McCullough offers that the branching bryozoan may be in the genus Halophylla, which means salt-loving.
Partial on this.
Gorgeous. This sponge is playing host to a series of brittle stars. That's the sort of pinkish ones that you see, and crinoids. Those are the yellow ones. Two different classes of echinoderms. Crinoids are suspension feeders. They're holding their arms up in the water column. They're using their little tube feet to flick particles out of the water and into a channel in the arm, whereas the brittle stars may be feeding on stuff that gets caught on the sponge, or maybe with their spiny arms can trap something into water column themselves. In the upper left, as you were zooming in, I think I saw a black snail. Oh, it's a hermit crab. I see. Okay, it's a hermit crab. We're fine with that.
Yep.
And then there's an Acanthogorgia coral here. It's the first one of those of the day. That's the beigey octocoral that you see on the left, and otherwise you see the very large holdfast of this sponge. So I haven't seen, Joanna's still talking about the Hertwiggia, but I haven't seen her give me a name for this one. I suppose I could look this up myself.
A little more further down, white. I was going to come back and grab that whip above him. Yep.
Oh, and this is there's a hydrocoral. That's what this white thing is that's branching, very thick branches, and you see all these individual, it look like filaments arising from it. I believe those are each individual zooids, dactylozoids. Probably pack a pretty good sting. If you've heard of fire coral or lace coral, this is basically a deep sea version of that. So in a different group of the cnidarians compared to the stony corals, which is where the cup corals we've been seeing and all this rubble comes from, and the octocorals, which are more like the sea fans. Faria. So Joanna says this is another Faria. We've now seen a couple of Faria. They either have very different morphologies, or we're seeing them at such different stages of their life that we can't recognize the transition between them. Oh, is this a bamboo coral finally coming into view? Or is it going to be a primnoid? I think it's going to be a primnoid, a really tall one if it is. Wow, I don't know that I've ever seen one this tall. But there's something about the regularity of the diameter that tells me it's primnoid. Yeah, It absolutely is. I can see all the polyps are rising from a whorl. They're pointed down. It actually looks on the left like they're pointed up, but that's only because this colony, we haven't seen the whole thing, is going up out of frame and then it's curving and coming back down. Oh, that's remarkable.
Take a little more.
So the family Primnoidae is another one of the three really common deep sea families of octocorals. Hundreds of species that have been described in, I think, more than 60 different genera. And the only unbranched ones that I know of in the Atlantic are Convexella and Candidella. And there is one that has the name Candidella gigantea. And to me, this is a pretty, for this kind of octocoral, it's pretty giant. But I have a feeling, my vague recollection is the name gigantea referred to the size of the polyp and not the colony. Eric Fielding's asking, what is the stuff that looks like cobwebs? Great question. We ask it very often.
Try to pirouette.
Do you have the lasers on?
Lasers on now.
Thank you. Those are 10 centimeters apart, and it's really hard to get the lasers with respect to the vertical distance, but we can do that in the post-dive viewing of the images and try to get some sense of how long this thing is, but it's a good one. The corals are definitely picking up as we get upslope, as are actually the sponge diversity. We're seeing more different kinds of sponges. Again, another good sign as we get to the top of this ridge that the currents are changing.
Copy your full line.
Yeah, Les Watling agrees with me that this is the tallest unbranched primnoid either of us have ever seen. We've both done an awful lot of deep sea dives and watched these videos, so that probably means something.
Partial sea star. Maybe a dust. Yeah, that's an octocoral. I think.
Truer words were never said, Scott. That probably means something.
I am profound at times. I think that was another of the hydrocorals, actually, that we were imaging just below that sponge. Look how fat the branches are. Yeah, and that's got a really hard calcium carbonate skeleton, so it has a skeleton very similar to the stony corals.
Now those hydrocorals, do those tend to always be? I feel like every time we see them, they're pretty small. Or have we seen any larger ones?
No, we haven't. I'm trying to think of an example where a hydrocoral is large, and maybe in shallower water, maybe up in the Aleutians, but in general, at least the depths that I'm more familiar with, yeah, they're pretty small. They tend to be pretty small.
The outcrop coming up to your
another stock crinoid here. Looks like it's overgrown with snail fur, Hydractinia, a hydrozoan. We did get a collection of one of these yesterday, and I can already see from a distance. So every one, and I've seen quite a few of these on this expedition, every one of these stock crinoids that we've seen has this overgrowth of hydroid on its stalk. So the business end, the feeding end of the echinoderm are those whitish branches that you see on the far right, and then the long stalk that's attached to the substrate is, well, it's its stalk. I guess it's in the name, right? But the pink stuff that you see is overgrowth of a hydroid, which is another kind of cnidarium. In fact, this is very closely related to that hydrocoral we just looked at. They're both hydrozoans, and those filaments that you saw extending from the hydrocoral, that's basically what you're looking at here. They're just a much larger version, and then you can see on the quote-unquote filament, in this case, it's the feeding business may have been tucked inside. We did see the stalk of a crinoid being overgrown by the purple octocoral earlier, but the crinoid was dead and there were no arms at the end of it.
Two purple whips at the top of where the white is.
By the octocoral?
Yep.
Or the hydrocoral?
I can snap it from here. It might be good enough.
Yeah, if you want to snap that.
Oh, nice view. So this is a Plexauridae. We collected this, I think, on dive 1 or dive 4, where we had both colors, the blue and the yellow together. Gorgeous. This one looks like it has yellow tentacles, a blue polyp body, and then light blue main axis to the skeleton.
Yep, branched.
Really gives it a beautiful appearance.
Slight tilt up full screen. Slide down. There you go. And video's clear.
All right. Let him go. Come away. I'll come back over to starboard after this, Sean.
Yeah, cut that.
Is it me or are we starting to see more and more octocorals?
Yeah, so the suspension feeding organism way picked up now. So we've gotten high enough on this slope that there's got to be enough current and enough food that the community is changing from what we saw earlier. And I just had a question in the chat here from Anna Kolako, who's asking if it's a known symbiosis or parasitism, the association of those hydroids with the echinoderm. And to tell you the truth, Anna, this expedition is the first time I've ever seen that association. So I can't answer that. I would suspect it's closer to parasitism or commensalism, when one benefits and there's no effect on the other. In fact, I see one of those stock crinoids at the top center. It's kind of yellow, and there for a change, there's nothing on it. So I wonder if it's just those crinoids are so old, they're finally getting colonized.
I think I can see them on it.
Yeah, now that I say that,
come in.
You don't have to zoom to prove me wrong. Oh, well, they are. So there you go. Yeah, I said it, right? So yeah, it does have some hydroids there right up on the top. And actually, that's another interesting thing. I think most of the time when we see it, it's just the upper part of the stalk and not the lower part. In front, just out of frame, is a kind of ratty-looking black coral. It's Leopathes. It's missing most of its tissue. You see it's still alive. It has various brittle stars on its branches. I see some barnacles in there. So that's good. We'll probably see more of those. This is, if you haven't listened on the earlier dives, the oldest known coral and the oldest known animal, so far as I know, belongs to this species, or belongs to this genus Leopathes, aged at more than 4,200 years old. And just out of frame now on the upper right, we passed another fan. I think that was a Candidella imbricata primnoid octocoral that we were seeing quite a bit of yesterday, I believe it was. Yesterday and the day before. And we made a collection yesterday of an Acanthogorgia. And Chris, I don't remember if you were the pilot at that time. We grabbed a part of a skeleton, and it came up with actually 3 different corals on that one bit of skeleton. An Acanthogorgia, an anthotheelid, and a bamboo coral that I didn't even know was there. Beautiful sponge, and then maybe a Chrysogorgia underneath that
turn
sponge.
This a little bit to keep in view. We're all settled
take
out.
A snap off. Just let it come into frame.
Let's see. Yes, Chrysogorgia. This one, I believe, is a different species from what we've been seeing earlier. Chrysogorgiidae, these are the golden corals, the whole family. And there you go. You turn on the light, and there you see that gold skeleton, iridescent, not iridescent, but it's reflecting the the light back as gold, and it looks like, is that a shrimp in there, or is that a galatheid crab? Probably, oh, that was a shrimp, okay. And very often when we see the shrimp inside these colonies, it's often a female that has eggs. There's a galatheid crab or a squat lobster down below it to the left, that magnificent sponge. And note the difference when we have the bare, I shouldn't say bare rock, but we have this rock that's coated, and we don't have all the knobbly coral rubble on top of it. And I imagine that coral rubble is a lot less stable than,
you want to center it and fill
for
the
want of a
screen?
better word, I'm going to call it more sheety, smooth rock. I realize it's not a sheet flow or anything, but
You want it tight or no?
Yeah, we can. You want it?
this looks like it probably has scale worms on the inside of the sponge. You see the sort of yellow-orange, yellow-orange with a bit of pink there associated with it. I noted that on one of the earlier sponges we zoomed in on. And we collected some sponge yesterday, and I was pulling a few polychaete worms out of the inside matrix of the sponge. Beautiful.
Let's get the whole community.
So in answer to your question earlier, Ashton, about the hydrocorals, Mary Milan from Italy is saying that there's a stylastrid coral, Erinia dabnei, which can build large colonies and aggregations. It's found in the Azores region between 300 meters and 1,000 meters depth.
Became 5 degrees.
Says, I think it is an endemic species, but it was found south of Fayal Island.
Coral
here's an Iridogorgia right in the center. So we're definitely in a new region.
I'm just going to snap it real so come back around a little bit.
Ken Sulak is saying, oh, look at that behind it. Sorry, so we can see that Iridogorgia, this is that spiral coiling colony in the front. It's a member of the Chrysogorgiidae. Nice plexaurid fan to its right. But I'm looking at the thing behind it. I guess it's an anemone. At first I thought it was going to be, oh, it is. It's our first serianthid of the cruise. I can't believe it. Not only that, it's tall. So that brown thing behind it, that's the common name is a tube anemone. It's a serianthid, and you notice below the brown there seems to be a collar, and there's a tube that disappears into the sediment. And some of these species I've seen, that tube can be 3 feet long. That tube is secreted or constructed by that anemone with a combination of mucus, a specialized kind of nida that are only found in these anemones that it fires and binds up sand and mucus into these tubes. They don't like being out of tubes. When the few opportunities I've had to collect maybe shallower ones and you have them in an aquarium, if you cut them out of the aquarium, they will immediately start making a new tube.
And can the head of that recess into the tube?
Yeah,
or?
absolutely.
Yeah.
And we've seen that very often. Anytime an ROV pilot tries to grab it, and it'll go in quick. And so that is for protection. So one of the ways that you can tell that it's a serianthid burrowing anemone is that cluster of fine oral tentacles that surrounds the mouth. So there's 2 rings. There's one on the outer margin, and then there are those smaller ones right around the mouth. Beautiful. I'm surprised we haven't seen more. Is there a sea spider on the base of it? The base of the tube? Sorry, Ashton.
No, go ahead.
I guess that's a sea spider. It's got too many legs. Is there 2 sea spiders?
1, 2, 3, 4, 5.
Let's try 30% of the extra light.
Great.
Not too far up.
We're getting some information from Tina Sacco. Yes, a tube anemone in the family. Oh, that's funny. Arachnididea. Sounds like a spider. Or, oh my goodness, I got to pronounce this. Batrunidiferidae. Batrunidiferidae. Just rolls right off the tongue. Cool. It's a pretty beautiful animal.
All right, I'll let that go. Yeah.
We haven't yet got the full screen.
Oh, understand.
Yeah, Ken Sulak is saying that he agrees we're seeing a major shift now in habitat context or biotope. We're leaving the sedimented substrate that is good for diverse fishes and moving on to the rock biotope, which is good for sessile, particulate feeding invertebrates, but not particularly good for fishes. So his prediction would be that we will either see fewer fishes or less diversity of fishes. Ken's been providing a bunch of information, but at the same time, that the activity is picking up on screen, so I'm not able to keep up with all the interesting
it's starting to level out
information that he was
would you feel good with like a 10-meter move?
Yeah, I think 10 goes.
Yeah, we had earlier seen a halosaur earlier in the day, and he says that the halosaur he saw there was a very, very gravid female. That means she was getting ready to release eggs, and they can shed tens of thousands of 1 millimeter yellow eggs. Those eggs are buoyant. Uh-oh.
Did we see that?
Sorry.
Upper left.
Maybe I should get you to read these, Ashton. Where did it go? The eggs are buoyant, rise to the sub-epipelagic zone, and hatch as transparent leptocephali, larval stages, so way up in the water column. So this is another way that the deep sea is connected with the shallows, and the shallows are connected with the deep sea. And this is a black coral again, and is it a small Leopathes, or is it a Stauropathes? Yeah, so this is a younger Leopathes. So earlier we saw that one that was really ratty, was missing a lot of tissue. This is a younger one. And it's kind of interesting. We were noting in some of the past dives how slowly they grow, and I said that they could increase in diameter by about 1 millimeter every 50 years. So when I say that's a younger one, it still may be, what, 300 years old? I'm just tossing out numbers now. Yeah, I mean, it's just getting out of diapers at that age. There's a metallic gorgia on the side of the rock there, several sponges, maybe a Hormathiid anemone, that large orange-pink one. Another one of the Faria sponges. Joanna noted what she calls an urchin sponge. I call the hedgehog sponge, possibly Polymastia corticata. And we did collect a sponge like that a couple of dives ago. It had a morphology that I'm not used to seeing. I do believe it's the genus Polymastia, but I don't know what species it is. Joanna says there must be loads of animals that are living in the fossil framework that we can't see. There's so much three-dimensional structure here. Oh, interesting. As near as Tina can figure, that that tube anemone that we were looking at has never actually been collected. And it's really hard because it retracts so deep into that tube. So here's a sponge we've seen in the past a lot, but I don't think we've seen on this expedition. See if we can get a name on this sponge. Most certainly in the Hexactinellida, the glass sponges. You can see how carefully the silica fibers have been laid down to create this intricate network. I noted a couple of anemones or zoanthids growing on it there. Oh, thank you. Conolasma coenoides. So the genus Conolasma, genus coenoides. There's our first Paramerciid of the day, and then that name is referring to that yellow fan-like coral that you're seeing down below. And let's see. Yep, I'm going to stick with that name. Possibly Muracides, I suppose, but at any rate, it is in the family Plexauridae, which I like to refer to as the deep-sea Gorgonian sea fans. And what I mean by that is the skeleton is protein. It's much more flexible than we see in the primnoids and bamboo corals. And it often has a pigmented tissue. Another large sponge. Several of bivalve shells look like littered down here on the bottom. There's a bamboo coral. So you note this whip is not as straight and smooth as that primnoid whip that we saw earlier. The polyps are a little bit larger. They're not arranged in whorls. This one you should be able to see the dark nodes interrupting the white skeleton at intervals. The hallmark of a bamboo coral. Let's see, where are the nodes in this guy? I'm not really seeing any right here, and I don't know if that's just because the tissue is so opaque or there's such a long interval between the nodes, which is possible. Now there, finally I see one just below that angle in the branch. There, you can kind of see it just the top of the screen. And then I don't see another one. Wow. So this one's got some very, very long internodes as near as I can tell. I wonder if there's one also at that bend, but that's interesting. Yeah, near the bottom. Agreed. It's the same sort of pattern as that upper one. It looked like there was one sort of crink in the skeleton between those two nodes. So Les Watling and his former student Abby Lapointe have just published a paper that came out, I think, in the beginning of July. And one of the genera that they describe is Taniostia, and that name translates as long bones. And the idea behind the name was how long those internodes were. So that's probably an Anthomastus, but we can check that out. No, it is not. What is that? That's like a file clam blowing a bubble? I wonder if, I don't know if that's a little tunicate or if that's an egg that's been deposited there. That is a clam. It's not really in the place that I would expect the file clams. I've been on the hunt for file clams this whole expedition. Don't really feel like I've seen it. Huh. Not getting any help on the red blobs. And I see Jean-Marc Gagnon has just joined in the chat. He's the resident expert on the Acesta file clams, so he'll let us know what he thinks of those. Single empty shell of file clam is what he's saying. Yep. Yeah, great. I would like to try that. I've been intentionally not asking you to sample anything for that very reason. I wanted to see you check to see if we managed to get anything. So thank you. Do you think you saw something on the canister? Which canister? Was that canister 2 or? Thank you. Yeah. Yep. Yeah, if it's not too far, let's see if we can get it. Because it looked to me like that was the larger piece of the two. Yeah. I agree with Copilot. I think. All right, that looks great. Nice job, guys. That was a tough one. Thank you. Mike Vecchione's giving his opinion that that red blob that we were looking at closely on that clamshell may have been a cerate egg. That is the egg of one of the Dumbo octopus. And he said it looked very translucent. And usually when we see those, they're opaque and white, and then they're secondarily coated in some other membrane. And he says it's possible that it was very recently laid and the outer coating had not fully hardened in the seawater. So that'd be cool. I've only ever seen those eggs deposited on corals, so it would be neat that it's on a different substrate. It'd be even neater if we saw the octopus.
And so we're just going to be hovering here for a moment while our ROV crew does a shift change. And so the ROV pilot, co-pilot, and navigator are going to give kind of a situational report to the folks taking over for them.
Hey, pilot, welcome to the chair. So we've been trying to motor up this slope a lot of the day. We haven't been seeing much, but then in the last half hour, we're seeing quite a bit. It's pretty clear to me we're not going to reach the top of the crest by the end of the dive. So if we do continue to see this more diversity, we're happy just to sort of look at what we have and not zip by it. Thank you. Oh, I'm sorry. I'm muted. My bad. Can you zoom in center of the screen? Yes. Yep, very small blue-gray. And it's a rock coated in a probably encrusting sponge. Okay, not at all what I thought it was. Thank you. I see a black coral Stichopathes come into view, orange coil right at the top, 2 Iridogorgia colonies. I assume it's 2. I suppose it could be 1 bifurcated. That would be unusual. Not sure what's going on right there. Probably another colonial tunicate hiding underneath that sort of ring of dead coral. There's a shrimp at least in the left of those 2 Iridogorgia. I don't see one in the right. Nope. No, this is good. Thank you. Maybe the first Anelopsamia of the day. That's the very bright yellow hard coral, stony coral that you're seeing in the upper part of the frame here. We've seen lots of that in the past week, actually. We've seen quite a bit of it. In fact, this one looks like there's yellow and white. I wonder when we look at the zoom here if there's going to be some overgrowth, or maybe it's just part of it's dead and there's sponge overgrowing it. Okay, yeah, there we go. So I think this whole skeleton that you're seeing going all the way off the screen to the left is this stony coral Anelopsamia, and only the right part of it is alive. It's got this thin veneer of yellow tissue, and then the white that you see on the sort of left of center, that looks like it's a sponge overgrowing, and then at the base there's a cup coral, and then the brownish part you see, all the tissue's dead, and it's just encrusting. There's probably a combination of bacterial action, settling particulates, and so on. A little bryozoan fan that's waving down below it, a couple of brittle stars on the branch. Very good. So I wonder if a lot of the rubble that we're seeing may be from species like this. You can see the branches on the right side are broken. Oh, good. We're seeing also the Iridogorgia on the lower left. Definitely 2 separate colonies and not 1 branching one. Oh, we're looking at a pillow, I think. An ancient pillow.
An ancient pillow dissected by fault motion.
Yeah.
Dun, dun, dun.
Whose fault was it?
The Earth's.
Oh.
Yeah. Oh, plate tectonics. You've wronged me again.
This may be
He's deep in thought.
yeah, starts with a D. Maybe it's actually another one of the tube anemones. Yeah, it's probably another one of the tube anemones. I seem to remember having this conversation before that I thought tube anemones always had to be in soft sediment. But Tina Molitsova, who's in the chat room here, corrected me that no, sometimes they grow on hard rock and they have a relatively short tube. So that's indeed what this is. Again, you can see that central ring of shorter tentacles around the mouth. And then the larger ring of tentacles are on the edge. So our second serianthid of the day, and maybe only our second serianthid of the entire expedition.
Now, is it the lighting that's causing that color, or do you think that this?
No, they have this deep purple color.
Yeah.
Very interesting. Yeah, there we go. Getting a good view of the oral tentacles folded down over the mouth. They're a little bit longer. I wonder if it recently ingested something.
I would call that color midnight sky.
Well, that's good.
Yeah.
Gorgeous. Science is happy when video's happy. Vagabond tube anemone is the way it's being put in the chat room. That gives me a little extra time to think of the name of the one I couldn't remember. It's got these very, very, very long tentacles.
Is it tentacular longia?
That wouldn't be bad. If we could, you don't mind, pilot, how about a zoom on the brownish bulbous thing coming into frame from the right on the top of that cluster of dead corals? Yeah, just to the right of the lasers. Thanks. I think it's that same quote-unquote pineapple tunicate that we've been seeing since we first got down. But I just want to verify it. It disappeared for quite some time along the slope, but now it's starting to reappear again. Yeah, it's too bad Joanna's not here anymore. So this is something, when we first saw it, I called it a sponge, and then we all decided, no, it's probably some kind of tunicate. And we tried to get a collection. It was on kind of rubble like this, and we grabbed it. It did look pretty gelatinous, so pretty certain it is a tunicate. I just find it interesting that it seems to have a hollow interior. I'm not sure why that would be the case. Most tunicates I'm familiar with would have the incurrent and excurrent on the same side, with the exception of the pelagic salps and or the pyrosomes. And this almost looks like it has the construction of a benthic pyrosome, which means that the water is being funneled from one side onto the inside and then would eject through that, for want of a better word, osculum. Very interesting. It's a real shame we weren't able to pick it up. Okay, that's good. Yeah, and Tina is saying that the Serianthus is normally deeper than 3,000 meters, so that was kind of interesting. Mike Vecchione says this has been a very tunicate-y dive. I agree with him there. And Bryozoans, Mike. Both taxa that, you know, we regularly see, but not in the numbers that we've been seeing today. There's a black coral Leopathes coming into view. Center left. Some big sponges. Relicanthus, that's what I was trying to remember. It was a pretty beautiful scene here. Several different kinds of sponges. I think I'm seeing some Candidella primnoid octocorals. Oh, here it looks maybe like a new black coral for the day. This feather-like colony that is extending from the left side of the rock outcrop here, left of the sponge, kind of grows like a feather. One main axis along the center, and then side branches coming out of each side. We call them, in the black corals, they call those pinnules instead of branches. On the other side, I think I see something like a Victorgorgia. It's bright purple, just disappearing off screen to the right. And I think that thing that kind of looks like a broom at the top is a hydrozoan. Hydrozoan or a bryozoan. There's a cup coral. So yeah, indeed, this is a Bathypathes black coral, and I think I've been seeing Tina identify this as Bathypathes pseudo-alternata, which is referring to the almost alternating way that the branches come out on either side of that main stem. But this is, again, a black coral. The black refers to the internal skeleton, which is made of a protein that forms a pretty dense structure. It's quite beautiful when it's polished. The black corals can have all kinds of different outward colors because the tissue takes on a number of different colors in different species. There's a very small colony of Candidella above it. That's the white octocoral. And then there's another crinoid, stalk crinoid, that once again has that hydroid on the stalk. And then, Pilot, why not zoom in on that thing that looks like a broom or a feather duster? Thanks. Let's see if we can find out if this is a hydrozoan versus a bryozoan. Oh, excellent. Oh, fantastic. Yeah, bryozoan, 100%. A really good view of the lophophores there. So the lophophore is the name for the ring of tentacles. It's kind of like a horseshoe-shaped ring that surround the mouth. They're ciliated. They generate a current. And you can see it's a colonial organism. There are literally hundreds of those zooids on there. That was great. 2 cup corals. Ken Sulak is got to leave us, our fish biologist. He thanks us for the voyage to the Ridge 2. Says we've had a nice illustration of total faunal shift at the same depth from sediment-dwelling community to the hard rock community, and he thanks the pilots, camera people for the super fish imagery. First subsea images of at least 2 species. So a little baby Iridogorgia colony. And then there's this brilliant purple thing down here. There's actually a couple of them down there. Earlier we had a look at one and we decided that it was the stoloniferous type. Those are the type that overgrow the substrate in kind of like a mat. If you look at all these octocorals that have the branching fan-like appearance, and there's a whole bunch of polyps, all of those polyps are interconnected by what we call senenchyme, and the digestive tract is continuous between polyps. A stoloniferous form has that same senenchyme, but instead of enclosing a skeleton, is just basically laid flat on the substrate. And that substrate could be a rock, or it could be a coral. In this case, it's coral rubble. There you go. And this has this spectacularly beautiful purple color, which is it stands out because it's unusual in the deep sea. There aren't many species that have this sort of color. And I noticed another one of those little globular sponges with the chimney that comes out of it. Kind of cool. And as I said, I picked some of those that were less than a centimeter in diameter off of some of the rocks that Ashton collected earlier this week. Oh.
Well, we're coming up on a depth of about 1680. I think our targeted shallowest depth was 1610, but that was a pretty long track. So I think we made pretty good distance. And
yeah, we did.
A pretty good coverage of depth.
It was a hard wall to climb. It's so steep. You have to be really careful with these vehicles. We don't want to slam the ROV into the wall, and so we approach that wall very carefully, work up it at a safe pace. Good communications between the bridge of the ship, navigator down here in the control room, and between the two ROV vehicles themselves. Iridogorgia just coming in center above the lasers now, one of the most beautiful of the octocorals, that sort of graceful spiraling pattern. And the branches, I'm pretty sure most species only come off of one side of that axis, but because the axis coils, you get that sort of fireworks effect. Someone once described it as a fireworks coral. I think it's a great name if you see it from the side, because those side branches droop down, and with all the little polyps on it, it looks like an exploding firework. Which takes me back to the very beginning of this cruise, because I got on the ship on July 4th, and we had a spectacular fireworks display in Norfolk that I got to watch from the deck of the ship. And I noted earlier, many times these Iridogorgia have a shrimp in the branches, and often the shrimp is a female carrying eggs. This one does not have eggs. The eggs are normally a bright purple color, and they're held underneath the abdomen, which is basically the tail part of the shrimp. And you can see that this individual does not have those. In fact, Les Watling is in the chat, and maybe he can confirm if this is a true story I'm telling or if I'm making this up. But we were on a cruise together in the Bahamas, and we were collecting an Iridogorgia at about 1,600 meters depth, and we clipped off the top, and in so doing we disturbed the shrimp that was in there, collected it, and had occasion to go back and saw a shrimp on it. And so we think the shrimp swam away and came right back to the colony.
And you know that because you verified the shrimp's identity. You asked its name.
Checked its card.
And said, I'm Sally the shrimp.
And so that exactly is the point. Was it the same shrimp or a different one? But we weren't sure. Well, why would a different one in the time? I mean, we're talking about, within a 15-minute time frame.
Oh, I thought you were talking like a year later.
No.
Oh, okay.
No.
I think that's likely.
Yep.
That's good evidence.
The yellow up there, I guess that's a sponge overgrowing dead coral. This really interesting matrix feel to it. Maybe we can zoom in right at the center top there, Pilot, the white branches that are standing up with relatively large polyps. I'm wondering if those are barnacles on a skeleton of some kind. I can't quite tell what it is, but whatever it is, they're fairly large. So that's why I'm guessing barnacles on a otherwise dead coral. Megan McCullough is offering an ID of that like feather duster bryozoan that we saw earlier as Celeria. Thank you.
There we
go. Barnacles on a bamboo coral skeleton. It's alive at the base with bamboo coral polyps, and the top has all these barnacles. Yeah, go ahead.
Did we do our third Niskin, our special Niskin?
No, we haven't.
All right.
Did you think this is a good spot?
No, I mean, up to you. Just try to remember to do it.
Yeah.
But wherever.
Why not? Because we're going to be doing another one in half an hour, so why not?
Yeah.
I think I'm glad you said that. So, Pilot, I don't know if you're copying that. Let's go ahead and trip Niskin number 3 here, please. Thank you, pilots. Thank you, Ashton, for reminding me about that. We just took another water sample for eDNA, and we take 5 water samples during a dive, and our middle one, number 3, is arbitrary to where we are anywhere on the dive. If we see a high diversity or something of interest and we want to see if we can capture that DNA in the water sample, we'll go ahead and take it. And then we take one just before we leave bottom, and since we'll be leaving bottom in about half an hour, we might as well take one here now and then compare it to whatever we see at the time. And then our last sample will be taken in the deep scattering layer, which is up in the water column where our sonars reveal there's a concentration of fish and zooplankton. And so we take it there to maximize our chance of seeing something. Looks like the sponge has a bunch of associates on it. The one that's just coming into frame, upper right into the center, I suspect it's going to be covered in zoanthids. Right above the lasers, right in the center, the biggest one. Thank you. I'm looking at all those dots that are on the lower half of it. Yeah, there you go. So you see there's a whole lot of polyps that are growing on the surface of this sponge. Those are zoanthids, a type of cnidarian I really don't think I've talked much about today. These look to be solitary ones. By that I mean they don't appear to be interconnected to one another. They look a lot like anemones, but to my trained eye, there's enough of a difference that I know that they're, well, I have a suspicion that they are zoanthids. I shouldn't say that I know. I don't know that they're not anemones, but based on experience, it's more common to see zoanthids. There's some subtle differences between them. Zoanthids are very commonly associated with other organisms, or at least there's a group called the epizoanthids that live on sponges, live on corals, live on crabs, and so on.
What's your favorite zoanthid?
My favorite zoanthid, what's it called? It's called Bubba zoanthus, I believe.
Oh.
Yeah, let me look it up. And it's one that's a specialist on bubblegum corals, which is how it gets its name. I think it's Bubba gummy.
You know, when you Google zoanthids, you get some really trippy Google images.
Do you?
Yeah.
I think if Les is in the chat room, he knows that genus too. How am I going to find that? Let me see. I'm going to find that name for you. It's a crazy name. In fact, here's another interesting fun fact.
All right.
I'm pretty sure that one of the co-authors of that genus of zoanthids was Arvin's advisor.
Oh.
Ties all together.
Wow.
It ties all together. Now I just got to get the name.
That's a level of inception.
Exactly, yeah. Sometimes I do think I'm still asleep. Is it Bullygumma? Bully... Daphne just Googled zoanthids following your lead.
Yeah. If you're trying to picture a zoanthid, try and picture the realm of Dormammu in Doctor Strange. Oh. Well, it sounds like we've got about 25 minutes left on our last dive on the Azores Plateau, ending this leg that's mainly been focused on the Mid-Atlantic Ridge. We've done about, 2 dives on the Azores Plateau. One, maybe just the third one might have just kind of just on the very distal region of maybe the Azores Plateau volcanism. We're currently about 150 kilometers west of Horta. We dove to a depth of about 1,930 meters, and we've slowly been making our way up. We're at about 1,670 meters below sea level right now. This is dive number 10 for us. It's been a solid, fun-filled packed 10 days, and a solid, fun-filled packed 10 days before that of transit.
Yeah.
But I learned a lot about the Milnes Seamount, which we did not get a chance to dive on, unfortunately, because of weather.
I learned a lot about rescheduling.
Yeah. It's been a harrowing effort to get here.
Yes.
But at the same time, we also get here in a lap of luxury and comfort unprovided to ocean exploration 100 years ago.
Exactly. Yeah. Right. We are definitely spoiled relative to the Challenger expedition.
Yeah. I had shepherd's pie for lunch and some chocolate for lunch and some more chocolate for lunch after that, and a cup of coffee I didn't have to brew, and followed by a double espresso I didn't have to make. It's an easy life. We've seen some amazing geology and amazing biology along the way. Notice I put the geology first, though. I never thought I would have the opportunity to be part of an ROV expedition that dove right in the axial volcanic region of the Mid-Atlantic Ridge, let alone on a hydrothermal vent field in the Mid-Atlantic. Seeing that giant block of pyrite or chalcopyrite or pyrite in the beginning, that giant golden nugget, which I still wish, it had a little fracture to it. I wish we kind of could have broken and taken apart, because I wanted to know how pervasively pyrited it was through, but, well, amazing. I got some lovely ROV footage of what pillow basalts look like after they've formed and weathered a bit, and some pillow breakouts that I can show some students when I get back. I know I have enough knowledge now to know that I shouldn't know anything about subsea life. So I know the next cruise I get on with a biologist, I'm going to start being like, that's an octocoral.
Sure,
octocoral.
just throw out names.
Yeah. That's,
what are they going to say?
Yeah.
We can just, close the chat. Oh, here. Maybe Les Watling is responding to me in the private chat. Is that it? Nope. He wants to know about my flight home.
No.
Les, I need to know the name of that zoanthid. He doesn't always have the audio on.
And these series of dives have given me a much stronger appreciation of how important the geology is to providing a home and base for the benthic life on the seafloor. I knew it was important for me personally as a geologist, but the interaction between the geology and biology I've really come to appreciate over the last 10 days.
It's interesting. We have two different corals in the family Chrysogorgiidae, that big tubular one that was just going off the screen to the lower left, and then this Iridogorgia here. So two different genera, but you start to see how they're connected. They both have those really delicate polyps on really thin branches, but the branching pattern itself, the construction of the colony, is very different. There may be some other characteristics at the microscopic level that differ among them, but if you look at their DNA, some other characteristics, you can reconstruct their evolutionary history. And one of the things I find so remarkable, among the three large families, I've said this several times now, the bamboo corals, the Chrysogorgiid corals, and the primnoid corals, each of them traces back to a common ancestor, all three of those families together, and then the families diverge. And then there's been a huge diversification in the deep sea. There's all kinds of species and species diversity within each of those three groups, all in this environment, which tells you there's something they must be responding to. And I just find that amazing, that we can have so much diversity coming from a single ancestor. Yeah. Oh, that was... I was going to say, yeah, Les is calling it Bologumma zoanthus as well, but that's the name I typed in. He's going to find it. The little yellow colony next to the Iridogorgia. Sorry, James. Ashton and I were waxing poetic there, and I missed it. I have seen some Acanthogorgia today, so it may have been that. That may have been what caught your eye, which wouldn't surprise me since he's doing his dissertation on that group. Sure.
We're going back to our instant replay. It's one of the benefits we have.
Yep. Not really. No, unless he's talking about that. That's not little. He said a little yellow branching colony on the right side of the Iridogorgia that we passed. Oh, maybe, yeah. Yeah, sorry, James, we're not going to have a good enough view of that. Here's an example. We saw this the other day of this yellow sponge that overgrows this other sponge. You can see the yellow on the side of the large yellow plate sponge. That is like a bugle sponge. Pilot, can I challenge you to a collection? Can I challenge you to a collection? So not here, not this sponge, but if you zoom out and look to the left, you're going to see a whip going off to the side. You see it looks like a little hook, top left off the screen. Yeah. So let's image it first. I think it may be the same primnode that we saw down below, and there was a lot of discussion of it in the chat room because it's the longest one that we have ever seen. And so we aren't certain if it's a new species, a different species, or just for whatever reason in these current conditions, it allows the species to grow taller. So what I'm going to suggest is if you could just clip the top 6 inches or so and then probably suction it, I suppose would be the easiest, but I'll let you decide if it should go in a box or suction. In fact, there might be another one right over there. Yeah,
oh,
there's
wow.
a second one. These ones aren't as tall as the ones that we saw further down the slope.
And there's the Oreo.
Is that the first Oreo of the day?
I think so.
Yeah.
Wide open.
I'm sorry?
Bio boxes are wide open.
Yeah, can you believe it? We've been zipping up this slope. So this is indeed the target. Thank you. Was that in inner or outer? Thank you. Yeah, pilot, that was very nicely done. Thank you. Sometimes I hate this computer. Oh, come on. We have 10 minutes left in the seafloor portion of the Voyage to the Ridge 2. Be about an hour as we move up through the water column. We'll be taking another eDNA sample, and who knows what we'll see in the water column. So we'll have a nice final look at this ridge slope, about 150 kilometers west of Horta, I guess. Here, I finally found it. I said it correctly, but I couldn't spell it right. Bulla gummy zoanthus. The name refers to the affinity of this zoanthid genus to bubblegum corals, with bulla and gummy being the Latin words for bubble and gum, respectively, and zoanthus a common ending of genera names in the order, historically referring to the flower-like appearance of the animal polyps. There you go. Despite the fun name, it's unfortunately basically a parasite that overgrows bubblegum corals. This is a Metallogorgia down below. Les Watling suggests this might be Iridogorgia portalizii, and we made a collection of one of these a couple of days ago, and the collection was important because the type specimen has been lost. The type is what we call the material that's deposited in the museum and used by the species author to describe it, such that anyone can go back and reference it in the future. And so there's been some question about what it looks like, but he said that the specimens that we're seeing right here have a colony shape becoming narrower toward the top, reflect the drawings in the Blake expedition reports, I think from the, I don't remember if those are the late 1800s or early 1900s. Very nice colony of Leopathes coming into view, this black coral that grows so old. Anemone, Bathypathes black coral, one of Iridogorgia, presumably portalizii, Metallogorgia. So as we're scaling towards the top of this ridge, excuse me, we are seeing more suspension feeders, more corals. If we get, sorry, pilot. Yeah, just go over towards the upper right there. Upper left. Yeah, the other right. Yeah, those two colonies, especially that bottle brush that seems to be forking. Thank you. We're seeing a lot more suspension feeders and larger suspension feeders. So I really think that if we had gotten to the top of this ridge, and we're not that far below it. What are those contour lines? 10 meters, you think? Yeah, 1,700, yeah. So we're only about 80 meters, I think, below the top of this ridge. Maybe less if we don't go to the high point. So I think what's happening is the current is probably coming from the west and swooping over the top of this ridge and coming down. And so the corals that are closer to that upper surface are getting the benefit of what's kind of sweeping across, as opposed to the ones that were further down slope. And that's why the abundance and diversity is picking up. So I would suggest to anyone who's interested in these kinds of communities and wants to learn more about them here on the Azores Plateau that they target the top of this ridge feature. I'll bet it'll be quite interesting. We're looking at an interesting Chrysogorgia here that forks when it gets big, and I know that there are some Chrysogorgias that do this. I wonder if Les Watling is still around, if he can say... No, the lasers are on it right now. Yep, you're there. Thanks. It looks like it's almost 10 centimeters in diameter, and by diameter that would be the width of all the branches. The axial skeleton itself is really small. And then isn't this nice for Chris Ma? There's a massive sea star at the base of this colony that we can look at, so you can have one more sea star as the expedition ends. So this Chrysogorgia colony, all that fuzz going up, that's a series of branches. There's lots of branches in this thing, and the branches further bifurcate. There's bifurcation, bifurcation, bifurcation. But you see they only go out a certain distance, so it gives it this more or less even diameter all the way up to make it look like a bottle brush or a tube. But what's interesting about this one is that then the main axis forks near the top, and so you had those two major branch points up above. And as we're zooming in, the yellowish coral in the foreground, that's an Acanthogorgia. So there you go, James. One more for you. And we're looking at this, what I believe is a goniasterid sea star. And again, I'm looking at the large plates that line the outside margin of this sea star. And I'm pretty sure that this is one of the group that is a corallivore, feeds on corals. Chris suggests Plinthaster, Plinthaster. 2 minutes to go in our dive. We're looking at a Candidella, the white coral down there. That's Primnoidae, and I can see that it has its characteristic associated worms. They are in the genus, or at least they used to be, Gorgonia pollinoi. There may have been a recent paper that did some modifications there, but that name tells you Gorgonia pollinoi. It's a fusion of Gorgonian, which refers to the coral, and pollinoid, which refers to scaleworm, and it tells you that that group of scaleworms is specialist to live on these octocorals. This Chrysogorgia we're looking at, you notice you see a lot of very wide balls relative to the size of the polyps, and that's because it's in a reproductive state. Another look at the stalked crinoid behind there, once again with its hydroid fur on it. Overgrowing hydroids. Yeah, so this polyp is ready to mate. It's swollen with sperm and or eggs. I suspect it's one or the other. I suspect this is a male and or a female colony. I don't think they're hermaphroditic, but I don't know which one it is, and I don't know how we tell from looking. Great stuff. Shows me that colony is very healthy. Obviously, it's quite tall. It's doing really well. It's in a good position, getting access to lots of food, and this year it's getting lots of food such that it can produce a lot of gametes. That's a significant input of energy. So, pilot, before we leave bottom, let's trip Niskin number 4. Thank you. Well, gang, I hope you enjoyed your tour of the northern mid-Atlantic ridge, or at least as much of the northern mid-Atlantic ridge as the weather and sea state would allow us to provide for you, and here in the Azores Plateau. Ashton and I and the rest of the team have had a good time bringing you these images, worked hard to get them, but we all feel a sense of accomplishment when we see such fantastic images from the seafloor. We see so many new things, get these great new collections of species. Have all this participation from shore in our chat room, our scientists, and those mystery holes, which has really generated a lot of excitement in the general public. And I personally hope that people who saw the mystery holes are tuning in to now watch our dives and being introduced to all the other great things that are here in the deep sea that are perhaps a little less mysterious, but no less interesting. We've seen some habitats where we just had an incredible diversity and abundance of corals and sponges and all of the organisms that depend on that biological habitat. Exquisite. And then we've had, much like the early part of today, stretches of the seafloor where the density has been really low. And it tells us that not every place at the bottom of the ocean is the same. There are various characteristic factors, physical oceanography, food input, supply of larvae that control what lives where. And it means we really have to pay attention to what's going on at the bottom of the ocean and do our best to protect it for their sake and for whatever value it provides to all of us. So thanks everybody for tuning in. As I say, we'll be traveling up through the water column for another hour, and we will take an eDNA sample up in the water column. We will then pull into port into Horta tomorrow morning, and we will be done. But next week, the voyage in what are we called? Voyage to the Ridge? Voyage to the Ridge 3, or as Ashton likes to call it, Voyage to the Ridge, Return to the Ridge, will begin. And you'll have 2 new leads, geology lead and biology lead, Joanna Javier, sponge biologist, Deb Glickson, a hydrothermal vent geologist, and you'll go to the southern mid-Atlantic ridge where there's a lot more known hydrothermal vent activity. We'll be visiting some fracture zones. We'll also be visiting some more sites around the Azores, so you get to see lots of cool sponges, corals, fish, geology, hydrothermal vent endemic organisms, great stuff. And they're going to be diving for 3 weeks, so potential for a lot more dives. Thanks a lot, everybody. Scott France signing off for everybody.